let me welcome you on the second day of the 8th Advanced in Silico Drug Design workshop. And the first lecture will be on structures and how to get that. So we will cover the way which databases are usable to get the structures of ligands and macromolecules. But for the ligands, it will be only brief because you've heard it already from Johannes and from Warnek. I will be mainly speaking about the macromolecules and basically about how to find them and what to get, what you should be taking care about when you are looking into the structures when you are doing drug design. And I will finish with the aggregated view of PDBE knowledge base, because it's really a good resource where you want to see something, everything which is known about the structure so far. So for the ligands, depending on the scale you would like to get, you can start with the drugs itself from the drug bank. Then you can look into all bioactive molecules from Campbell or PubChem, or you can even take all of the commercially available compounds for virtual screening from the zinc. But as I said, this was already covered yesterday during the Johannes Kirchmeier lecture, and therefore I will be mainly speaking about the proteins and their macromolecular structures which are in the protein data bank. Here is a small reminder that the structure of nucleic acid for the protein is much bigger than the protein itself. Yeah, so this is the mRNA which is encoding myoglobin and in comparison with myoglobin itself. So you can see that there is quite a huge difference in the scale between these two structures. And that's probably the reason why the proteins are so much used as a, let's say, zipped information in order to generate a function. And the structure of the protein actually encodes what the function of the macromolecule would be. From the left, the first part is imatinib, which is binding towards the ABL kinase. And that's one of the first drugs which was firstly developed with in silico drug design. And you can see these type of kinases are quite important for the anti-cancer drugs, because you basically would like to stop the kinase in order to stop the division of the cancerous cell. And once you know the structure, you can try to generate the molecule which will be binding towards it, and as an inhibitor, basically to block this kinase from its function. But the structures can be quite huge, like this GROS barrel in the center, which is a chaperone protein, which has to be big enough, because as a chaperone, it has to be able to get some smaller proteins inside to help the fold. And on the right side, this is ATPase, and you can see that it's quite a big structure which is moving, and from that movement, it generates the ATP. So really, the structure defines what the function of the protein will be. And specifically, when we are looking for from drug design perspective, we are basically looking either on protein-ligand complexes on the left, because we can see where are the ligands binding, where are the functional sites. But also, we can look into multimeric states, because in that case, you can basically look into the interface between protein-protein and try to find out for instance, protein binders in order to develop the vaccines, antibodies, and so on. The problem is that to gather the structure experimentally is still quite expensive. This image on the left takes the proteins from the human proteome, which has at least some part of the protein in a protein data bank. And even now, after decades of working on the protein structure determination, we still have only 30% of human proteins with some known structure. And 60% is completely unknown from this experiment. As you will see during the lecture, currently we basically, thanks to the AlphaFold, we have those structures, and we now understand why we were not successful to reach them. But we generally doesn't have that much experimentally determined structures, as we have the protein sequences or even genomic sequences or metagenomic sequences, where there is 4 or 5 orders of magnitude difference between the number of the structures, of the structures towards the sequences. Because with the structures, we are approximately at something like 250,000 of experimental structures, more or less. Well, rather less, it's 230,000. But with the protein sequences, we are at 600 million. And if you are going for the genomic information from metagenomics, it's even 1 order of magnitude higher. So we store the data, the experimental data, in a protein data bank, which is the database of multiple locations. In Europe and Africa, we have the protein data bank in Europe, which is located in EBI in Kingston, UK. There is for Americas, there is RCSB for the major protein data bank, and there is protein data bank in Japan for Asia and Indonesia, and Oceania and Australia, and there is new PDB China, which joined the consortium, I think 1 or 2 years ago. And there are not only, this is not just the protein data bank for the protein models, but it also covers the experimental information coming from the X-ray, coming from the NMR, and coming from the cryo-EM. And it provides the data are the same in all those individual locations, and they are offering a little bit different analysis based on what those individual sites are specialized in. But there is one general deposition system known OneDepot, which is working for all, and the data which are deposited into one are then spread towards the rest of these databases. And this database was established in 1970s, and therefore is the oldest open data database for which we currently have. And it's some kind of a pioneer in open science. It works, everything in PDB is working around the PDB code. And this PDB code basically codes towards the structure which is deposited. And this structure is typically connected to some paper, but quite often it's not, because there were sometimes large screens of structures, and therefore there is no publications behind. It's just the data directly from synchrotron, for instance. And they can be, by this code, referenced by the number. You probably know that there are 4 letters of those, like 2 ENS and so on. But this we will since the PDB is here for quite long, we are running out of those codes, and therefore the new encoding will be PDB and 8 digits. Yeah? So it will be different. Also, we already ran out of the codes for small molecules, which used to have 3 letters, and now it will be 5. Yeah. This is how the old PDB format looked like, and basically you get the information per individual, each line was individual atom, and it was annotated by the chain, by the residue number, XYZ coordinates, and also occupancy and temperature value, like called B factor, which was giving some kind of an information about how much precise this position is now. This format was already abandoned, and for the new structures you can't get it from the database, which is trying to say to all of the simulators that they really should stop to use the PDB format and start to use the new MMC file, because this MMC file is extendable. It's still text file, so it's still relatively readable, but it's extendable towards all possibilities, and you can add new things into it. So for instance, currently we are developing a way how to encode also the channel information or tunnel information into the MMC file. We already have the better annotation of partial charges within the MMC, and we are running that on the alpha charges server. So it's possible to extend and expand this MMC file more easily than the previous version, where just movement by 1 bit completely destroyed the reading of the file. So how you can get the structures of the macromolecules? The major method still experimentally is Röntgen. It's X-ray crystallography, where you basically crystallize the structure and then you X-ray on it and get the coordinates. Then this is now revealed by the cryo-EM, which is giving that you are spraying the electrons on top of the protein in vacuum, and you get the shape of the protein. And since you are taking it from multiple orientations, you basically get the structure as well. Then there is a possibility to use the NMR to look for where the positions of the atoms are, but those atoms has to be somehow unique, and that's quite a problem when you are repeating alphabet of 20 amino acids where every atom seems to be quite similar, and therefore you have to tune the NMR in order to see the surroundings of the proton within each amino acid in order to be able to distinguish what it is. And of course, the bigger the structure, the more redundancy you get, and therefore it's much harder to get the structure. And we have also mass spectrometry or FRET, where you just get the information about the distances. Yeah. And last but not least, we have the AlphaFold. But the major lecture about the AlphaFold is on Friday, so I'll be speaking only shortly about it. So in crystallography, you basically take the protein crystals, which you can, according to my old supervisor, Professor Tomáš Opšil, you can distinguish protein crystal from the crystal of the salt by slowly touching it. If it was a crystal of protein, it will destroy. If it was a salt, it will stay, which is a nice way how to distinguish that you had a protein crystal, because you are losing the data at all. So to get the crystals of protein is quite hard and tiresome job. And even that, after you have the crystals, you have to run the diffraction experiment. And from the diffractions, you can count the map of electron density and build a model into that. This is still the first method which was used to get the macromolecular structure as shown here with Rosalind Franklin and Raymond Gosling picture in Nature, 1953. And below is the visualization how it looks like for a small protein. Yeah. The major information about the quality of the data is still the resolution, the R number, which is used as a prime information about the quality of the structure. And it really depends on the R number, on what you can do with the structure. On the left, there is a 3.5 map of photosystem 2. And you can see that basically you get the knowledge about where are the individual side chains, but within the catalytic core of this photosystem, you basically had no idea what is the inside orientation of individual parts, and you just get the information that it's there, but nothing about the information, how it is binding and so on. So this type of the structure is not good when you are doing the, for instance, molecular docking, because you don't have enough of the detail how it looks like. If the second part is the 2.3 map of photosynthetic reaction center, and in this case, it's quite nicely visible the position of individual atoms, and therefore it's possible to see that, okay, this belongs here. And it's always worthwhile to check the electron density, especially of the ligands, because that's typically the problem when you are trying to use the structure. You should check whether it's correct or not. And if it's the resolution is even better, like almost 1, then you basically are able to see everything, okay? Which is great, and you can quite nicely know how precise you are. In MolStar, it's possible to visualize the electron density within the protein data bank or within the MolStar itself. And it's relatively simple. You just start the volume streaming, and you see the electron densities, and there are 3 maps. The blue one is basically the map into which the model was fitting. The green map and the red one are the changes that don't agree with the model. So the green ones are that you have some additional electron density for which there is no model, and the red one are that you are modeling something for which you don't have the data. Okay. And it's quite good thinking to basically check the electron density of the binding site to see whether the position of individual atoms of the ligands are actually right and true, because they might not be. Yeah. Because while the protein itself is checked by the Ramachandran validity, stereochemistry checks, length of bond lengths and bond angles, that's all automatically checked within the ProCheck or MolProbity software, yeah, basically automatically within the software. But for the ligands, since every ligand is different, this validation is much less used, and there are quite a lot of mistakes in the PDB. It's starting to improve because now there are validation even for ligands. Like Validator DB is basically looking into the ligand's quality, but once you are starting with the structure and you don't know it, it's quite a good first thinking to check whether what you are seeing within the model is actually true at the density level. For the validation, as I've said, we are checking the R3. Therefore, you are looking whether the structure is true. If you just take some of the reflections out, you check the stereochemistry, you check the Ramachandran diagram of phi and psi angles, which will give you the information that the amino acids should be in those red areas or in the yellow ones, but definitely not in the white, because that's that would mean that there is some kind of a strain within the structure. And of course, it can be 1 or 2 amino acids, but not half. Yeah. Of course, the same thing is for the sterics, problems in the structure. Good that check is quite similar in this line as well. Additional problem which comes within the protein crystallography and from the crystallography part is that the protein crystal means that you have contact between individual cells, unit cells. Normally, because it's a protein crystal, there is plenty of water which is shielding individual macromolecules from each other. But they can have a contact. This is called crystal contact. And it can be similar to the protein-protein interaction. So actually, in order to generate a benchmarkings for the protein-protein interaction, it has to be checked whether this might not be the, just the crystal contacts, which is just artificial. Yeah. And unfortunately, sometimes it ruins the structure. So for instance, here, from my experience, is the cytochrome P450, which typically is quite close structure. But once it was crystallized in this type of dimer, where basically two cytochrome P450s were crystallized that they are touching each other, because they were really strongly packed to each other. The biggest trouble with this structure is that these are truncated structures, and both of those proteins are attached towards the membrane by N-terminal helical anchor. And once you add this anchor into the structure, they definitely have no chance that the structure should be looking like that. Because in this case, one of the N-terminal will be looking to the left side, another one will be looking to the right side of the picture, and there will be 2 membranes basically. So it's completely nonsensical in the sense of the protein membrane environment. And unfortunately, that's just an artifact of the crystallization method. So just even the experiment can have their own problems. Second method is NMR. In NMR, we are collecting the resonances from the radio frequency, and then we get the chemical shifts, usually in 2D or 3D even. And from that, we basically count the distances for some amino acids, and then we ran molecular dynamic simulations or AlphaFold these days to generate the ensemble of structures that fit towards the coordinates and constraints coming from the experiment. The good thing about this method is that it basically gives you the information of the solution, because you'd have no crystallization in this approach, and you can basically do with the protein whatever you can do in the solution. So you can add the inhibitor, you can add another protein to check whether it will bind, so the signal of the protein will change. So you can do this type of experiment, but in order to get the whole structure determination, there is a big problem that you are limited by the size. Because, as I said, you have 20 amino acids. Each of those amino acids, in general, would have the same chemical shifts, and you only see tiny differences, and it will smear, individual amino acids will smear into basically the same blobs, unless there is some specific interaction. So in NMR, we don't have the resolution. But we typically can check the other metrics, which is possible, and that's the class score. You can check the stereochemistry like Ramachandran, and you can check for the outliers. So basically, based on that, you can see information within the protein data bank, whether the structure is okay or not, and you typically get the which parts of the protein is moving and which is not, which is quite especially good when you have a disordered part of the proteins. And from the NMR experiment, we basically can see quite a lot of data based on the NMR parameters like shifts, couplings, and relaxation rates. You can look for the hybridization or covalent, and you can derive secondary structure, interacting distances, torsion angles, hydrogen bonding, and so on and so on. And when you combine this, you can get the 3D structure. But of course, this, as I said, you are in solution, so you can basically tune and make the experiment with the protein, like the titration, for instance. The electron microscopy is the second most used method these days, and as I will show early on, it's getting quite a lot of traction. And basically what you are doing is that you spray your sample on the graphene grid within the electron microscope, and there you basically analyze the individual proteins which are there in different orientations, and then you generate the 3D volume map from those individual particles, and then you basically come up with the, you can fit the model into it. As I say, this is the data from today, and you can see on a logarithmic scale that all of the time the X-ray was giving the majority of the structures, typically 10 of 1,000 last 20 years. It's basically every year it's almost the same. NMR was giving only 1,000, and actually it's not much better. And its usage is actually less and less. But after 2009, 2010, there was a huge revolution in the resolution of the cryo-electron microscopy, which started to get the structures within the atomistic resolution. So we were able to see individual atoms, at least in the resolution of the, let's say, 3 angstroms, 2 angstroms, and so on. And therefore, the cryo-electron microscopy started to be useful also in a similar manner as with the, as was the X-ray before. And as you can see, we are slowly reaching towards the usage of the X-ray. So maybe the year 2025 will be the year when the cryo-EM will actually surpass the usage of the X-ray in order to structure elucidation. And part of the PDB is also the electron microscopy database, where the raw data are stored. And the electron microscopy has one big advantage, because it can give you the information about even the large complexes, like the viruses even. That structure of the rotating protein, that was done by cryo-EM. Yeah. So you can basically, and since you are looking into individual particles, you can see multiple different conformation of the same protein. And therefore you can then fit them by the RMSD to see the movies, basically. So it's really powerful method, and it's, as I said, it's getting distraction more and more. The next method is more of an additional one, because this is the mass spectrometry, where you basically make you ionize and accelerate the sample, and then you collect the individual fragments that according to their mass and and charge. And from that, you basically can come up with the information about this, what fragments it is, because you know the exact weight of the fragment, and therefore from the composition of the protein, you basically can come up what type, what is the composition from the amino acids, and if you fragment it more, you can even get the sequence. Yeah. And this is done quite frequently in proteomics, that you basically localize what type of proteins are there according to the fragments. But in the structural elucidation, you can use this with crosslinks, that you have a protein complex, you put there chemical crosslinks, and then you are trying to find the masses for those crosslinks, and you are looking for the environment of those crosslinks to get to the information of the distances of the specific parts of the protein complex. And that was actually used in a huge scale, because that's collected in a private database at the EMBL ABI. And they've connected it with the 25,000 mass spectrometers, and now they've used it as a information what type of the protein complexes in humans actually have the experimental information. So it helps quite a lot to find out which protein-protein interaction is not just a contact as in crystal, but these ones are quite strong, and they can be combined together with the AlphaFold modeling. And that's the last method I will be speaking about. And that's the method where you basically look for the sequence, you look for the templates out from the PDB references, and then you do the genetics search in order to get the covariant matrices that will give you the inference of the distances between the individual amino acids within the structure, and you combine this within the EVO former in case of AlphaFold 2, or in pair former in case of AlphaFold 3, in order to generate the structure which is quite good quality. And since we are here in the AlphaFold 3, you get here also a diffusion model that adds also small molecules, similarly as the Midjourney or DALL-E are generating the images by saying that, okay, I would like a coloring book with the cute cat. That's a typical query from my daughters. And I put this into the Midjourney, and it will use the white noise and the information from the denoising function, which was trained on a large amount of the pictures which had the information, this is the cute cat, this is the coloring book. And basically, I'm going backwards to generate the image based on this information stored. In this case, I'm just using it not for the information based on the cute cats, but in this case, the information is chemical, where I have the annotated structure with the annotated ligand, and that was noised. And if I change the equation and do denoising from the white noise, I'm generating the structure of the ligand inside of my protein structure. So the question is, how good are AlphaFold models good for drug design? Basically, the problem with the AlphaFold is that the general fold is perfect. At the level of the C-alphas, they are quite oftenly undistinguishable within the experiment. But for the side chains, it's not that stellar. And if you start to do the docking into the structure, or into the traditional model. Basically, quite quickly it became familiar that the success of docking, of redocking the molecules into the alpha-fold structure is quite limited. Here it's really similar to the traditional model, like from the homology modeling, and it was nothing stellar. While if you would be doing the redocking into the structure with a different ligand, the success rate was much higher. And that was also shown that the alpha-fold is not producing always the conformation which is actually good for the ligand, because in 30% of the structures it generates apo structures. In the meantime, as I will show in alpha foldology, there is of course a lot of the tweaks to generate multiple different conformers out of the alpha fold structure, and therefore it's much higher chance that you will get also the holo structure, which is the structure where the ligand should be. So the problem with AlphaFold is that the local side chains might not be exactly where you would need them for the specific docking task. So it's quite good to optimize first the side chains with MD before the docking. But there is a really interesting study from Brian Skoychet lab where they not do the retrospective docking, so basically redocking back into the AlphaFold structure and to say that it's for nothing, but they did the prospective docking. So they docked into the AlphaFold, and then from the selected hits, they analyzed experimentally whether those hits are good or not. And they found out that the drugs are that those hits are binding differently than towards the experimental derived structures, but they were able to reach even for less than 5 nanomolar binders than as with the experimental structures. So it's quite interesting that basically, even though that you cannot redock, you can find new things. And you can find new things and new structures in the AlphaFold database, which is run at the EBI. And basically what you get there is first you have here the model organisms, you get there the global health problems, proteomes, and also you have there the information about the Swiss prot. So basically you can reach quite easily more than 200 million structures and to look how they look like. So yeah, and the AlphaFold 2 did not have anything else, just the protein. The protein most of the time was holo, but without the ligand. So the ligand was able to put in from the crystal structures, and that was done by the alpha-fill. And the alpha-fill is basically putting the ligands from the similar crystal structures, and therefore you get the annotated AlphaFold with the ligands even from the AlphaFold 2. The good thing about the AlphaFold is that it gives you the information about where it's good and where it's not. First, you get the colors of the structure. Those colors are annotated with the PLLDT, which is a measure of the local confidence. If it's over 90, then this structure is completely right. If it's less, then from my perspective, it's more or less okay, but it can be movable. And basically, if I'm looking for my typical transporters, then the parts which is moving and opening between the structure, like the channel openings and closings, that would be in this area, like confident light blue, but not completely rigid. And then, if you are going below, yellow is low, and it's usually quite flexible. And then you get these lines almost, and that's very low. And it basically gives you the information. This is disordered region, and it says that the structure is wrong, but this part will be moving. So that's PLLDT. That's local confidence on individual amino acid level. But of course, the amino acids are connected to another ones in order to get the structure, to get the fold. And that's shown on the right with the PAE. And here, the matrix basically tells that this pair is positioned right or not. And if it's in green, dark green, it's really not visible. If it's dark green, then basically you have this part rightly, and therefore that's for this structure, and that's on the left corner where there is the combined structure together, and the rest of this of the structure can be basically quite flexible and movable anywhere. Then, when you are working with the multimers, then you can add 2 additional predicted values, and the PTM, which is prediction that you have the complex right, and IPTM, interface predicted TM, basically gives you the information if the interface you are having is okay or not. Yeah. And if we look into the PLLDT value over the human proteome, in resolved structures, it's almost all quite nice, and there is only a limited amount of the of amino acids with low PLLDT, while in unresolved, the majority was with low PLLDT. And if we looked over whole proteome, we basically can see that from 2/3, it's structured, but 1/3 of the human proteome amino acids are unstructured, which gives us the information about the measure of disorder, and the disorder is problematic for those previous experimental methods, because disorders is unstructured, and we are speaking about a method for structure elucidation. So let's move towards the disorder. Disorder starts to be more proficient now, and we've seen that 1/3 of the human proteome is unstructured, so it's really important. And it connected with the movement of the individual parts of the protein from some tumbling of individual amino acids or atoms even at the nano level. And if you are looking into the large movement, then we are speaking about the milliseconds or even seconds. The disorder in the traditional methods is unfortunately completely invisible. The only method which is able to capture the disordered parts was NMR. But even the NMR was not able to give you the information about how unstructured it is, only get you the information, okay, this part is unstructured, and there are multiple different ways how it can flow. Okay. We can get the knowledge about which parts are more unstructured from the temperature factors, for instance, from the crystal. And typically, when you have the temperature factor of the protein, then the binding site of the protein is really cold. It's really not moving. While the surface of the protein is typically more movable. And typically the N and C terminus are the most flexible parts of the typical globular protein. But not always it's the case that you have just this part, but quite often you have just one domain, another domain, and the part in between them is unstructured as well. So that's quite a typical situation as well. And the reason why we have the unstructured and some part of the proteins are basically interestingly disordered, they cannot take the order by themselves. They typically are interacting with the molecular recognition, so they are promiscuous, and basically they are catching protein here, catching protein there, and putting them together. They are quite often used for the molecular assembly, so by themselves they are not structured, but if you add additional proteins, they start to form something. And they also are working in an entropic chain activities like basically molecular hourglass and stuff like that. And there is a database where you can see for this type of behavior. It's called DisProt, and basically there you can look into the proteins and to find which part of those proteins is unstructured. But some information is stored in DisProt, some information is stored in PDB, some information is stored in, let's say, ChannelsDB, our own. And that's not really obvious way how to get to all of those information. And for that, there is the Protein Data Bank Europe knowledge base, which is aggregated view on proteins and ligands. Because in common PDB, you get the information for that specific structure. But in biology, you are interested in not that specific structure because you can have 100 of them already resolved from the multiple information with different ligands, for instance. So you want to know something about the biology behind. So you want to look into that specific protein and to see how this protein changes when it's interacting with different ligands. And for this is Protein Data Bank Europe quite good. It's there you get the aggregated view of proteins of all of their structures. These days also of the structures within the AlphaFold and Swiss model. You get the information about the small molecules, and you get the information about macromolecular interactions. So for instance, what I was doing for the Czech National Health Institute, when they were looking whether the COVID vaccine, the newest one, should be supported or not. They've asked how the interaction with the antibodies are changing. And basically what I was able to look at was into the Protein Data Bank Europe. There I looked for the spike protein. And there is such a huge amount of the spike protein together with some antibodies that it's quite easy to gather the information which amino acids are on the surface interacting with the antibodies. And this information I just connected with what are the mutants within the structures. So it's quite usable even in this type of the medicinal information. And there is plenty of additional functional information, and it's shown on the structure itself. And on the right, there is so-called ProtVista viewer, where for every amino acid, you have all of the information from multiple sources. You can see that there is plenty of information there about the flexibility, disorder, possibility that there is some kind of a channel from our channels database, whether there is some kind of predicted ligand binding site from page to rank from David Hoxa and Marian Novotný lab in Charles University, and more of those. And just recently, they added the web pages for aggregated view on ligands. So it's quite good to show that here at the drug designing workshop. And here you get the information for individual ligand, in which structures it is. And you can get the information also about the individual atoms within the structure and how it interacts with different types of amino acids, which is quite useful if you have something specific. But heme is almost everywhere. So still, it was quite interesting that one of those oxygens is actually interacting quite oftenly with serine. Or, as you can see, it's interacting, of course, with arginine and lysine, which makes sense because it's a carboxylic acid. So it wants to interact with something positively charged. But the serine was quite surprised to me. So, a take-home message. Each method has its advantages and disadvantages. And you have to check for those. So in X-ray or EM, you have to check for the electron densities, whether the model is true in that specific point of the structure, because typically the resolution which is stated is overall resolution. It's not the resolution on your specific spot where you are looking into the structure. There, that part you have to check for yourself. In case of NMR, it can give you more information about the unstructured part. And it gets you more technical information. With mass spectrometer, you can use the information from mass spectrometer to check whether the protein complex is right or not. And AlphaFold is these days easy to run. There is multiple places where you can run it. CollabFold, AlphaFold server from the Google, from the DeepMind itself, or you can use the installation at Matacentrum at Foldify, or you can install it on your own computer because it's open. It gives you the information about where it knows that it's right and where it knows that he's not right. And it showed that the PLLDT is actually our best disorder predictor, which we have right now. So the checking for the quality of the structure turns out that you can say which part of the structure is actually unstructured. And PDB knowledge base is a quite nice aggregator of those resources that gives you the information on one spot completely in order to see what you are looking for. It usually is there. And with that, I would like to thank your attention, and if you have any questions, please do ask.
What's actually how does AlphaFold behaves with antibodies?
Well, for the antibodies, the AlphaFold itself is probably not the best tool these days. There are better things like Bindcraft. Because AlphaFold, multimer can be used for designing of the antibodies, that's true. But if you want to design the antibody and design the sequence, then there is multiple different possibilities like neural plexer. There is Bindcraft, which seems to be the most successful one, or protein MPNN from the David Baker's lab, or Chroma from David Baker's lab. So, because if you are generating the antibodies, you are basically looking for the amino acid sequence that fits to the structure. And that's a different thing than the AlphaFold, where you are looking for the sequence and how it makes the structure. So it's the other way around. But these days, it's possible. Yeah.
Do you have any theory why the AlphaFold is preferring hollow structures?
Because we are preferring experimental hollow structures. Because we are doing the crystallization with the drugs inside, and therefore it learns that and use that in order to generate the hollow structures. But I know that, for instance, for the kinases, there is quite nice KinCore database where you can see the apo and hollow structures of multiple different structures. Because Roland Dunbreg, who is running this behind, basically attuned the AlphaFold that you can specifically hide individual inferences, and therefore you can make general more confirmations out of the output. And then, since he has long experience about finding out how the structure of the APO and holo look like in the case of the kinases, he basically is able to select the model of each. So KinCore has has complete human proteome kinome resolved in holo and APO structures.
Hell a lot of binding happens in disordered regions. Is there any light in the end of tunnel to tackle those regions?
I don't know. I'm not an expert on disorder, I must say. I'm just curious and cautious about that there is quite a lot of disorder, and this type of disorder can quite often organize itself at the interfaces. Like if you have a p53 protein, which is quite common protein in quite a lot of cancers, this protein is basically able to just structure itself to bind to quite a lot of amount of the proteins, and if something is wrong, then it's not possible for it to basically get those proteins together. So that will be hard. But yeah. But typically when it makes the interaction, then it starts to be structured. So maybe that's the weak spot, that you have to have some kind of structure in order to interact with the structured part. Okay. If there are no further questions, then I will ask Martin Lepšík from IOCB to continue with his lecture about the QM in drug design. Máš to na flešce?
Hello, can you hear me? Yeah, I hope so. Thank you, Karel, for, before you leave, for organizing this. And like this, I mean, the whole week, it's a great opportunity for you to learn really the wealth and breadth of drug design from different perspectives. So thank you for inviting me. I haven't come from far. I come from Prague, from IOCB. You can see, well, Prague you can recognize there, and IOCB you can see in the middle, where this is the old building. well, it doesn't show too much, but yeah, on the right you recognize the old building of IOCB, and on the left the new building of IOCB. So it is important, an important lesson that this was only able to be built by the money from a successful drug design project by Antonín Holý, a medicinal chemist with very deep knowledge in medicinal chemistry, but also he was able to learn a lot from virology and molecular biology, and thus with a bit of a lot of luck and a lot of courage also, he was able to drive several of his compounds to the clinics as drugs, and so he became a billionaire, and in the same time very yeah, down to earth man, and also our institute is very rich. It's always making some competitions with the Václav Havel Airport, who will pay the biggest taxes in Prague 6. So we are currently first or second place. So this is a great building for medicinal chemists. And the newest building is not shown here. It's been opened a few months ago, and it's cryo-EM. So
Photo is from the roof of the new building.
the photo, well, new building, yes, but cryo-EM is yet another building, and so it's only to show you that what Karel said about the hype of cryo-EM. So really, in these days, you really want to go for big systems, and thus cryo-EM. So well, I came here to save you time and money. So money, I will save you 213 or 14 euros, because you don't need to read this book, because I will tell you everything what is in there. No, I'm kidding, really. This is coincidentally the same title as my talk, but actually the book talks about very different things, and even we, our group or our approaches, don't have a chapter there. So I will rather talk about alternatives. So what is in this book is more approach of quantum chemistry from fragment-based. I will speak more about faster methods, which is semi-empirical quantum mechanics. So my name is Martin Lepšík, you heard that. The core of our team is there. It's called Computational Chemistry for Drug Design. It has been established in January 2025, so we are pretty young, but we have been in this area of drug design for at least 15 years. The group leader is Jan Řezáč. And now, let's see, so if I talk about every slide for 3 minutes, then I will probably not make it, so I may speed up a bit. And okay, such slides will be fast. So what I will tell you about. First, very generally about the roles of computer-aided drug design, then to our expertise, but there are also other groups within this area of semi-empirical quantum mechanical methods and scoring. Obviously, we are not playing some virtual games without any link to reality. So experiments still, you may say, bad luck, but we need to live with it. Experiments still are the cornerstone. But there are different experiments, good and bad, or else hazy or very unsure or reliable. So it's very difficult to choose and to choose well. So this is also collected in data sets. Then I will tell you a bit about different extensions of the SQM methodology and one case study of insulin binding to its receptor. So let's start. You may have seen similar graphs like this, which will tell you generally three things. The drug development is a process which is lengthy. It takes 13 plus years. You can see this cost per, cycle time years, so that there's like 1 or 2 or 3 years per each stage. Then it's a costly process, so you really pay a lot. On the left-hand side, in the brown, these still can be run in academic labs. On the right-hand side, where there is phase 1 and clinical trials, that's out of scope in terms of money, so it's mostly pharmaceutical companies. But if you are lucky, you can be bought by a pharmaceutical company or patent for a compound, etc. So you may also be part of the process, that you had the idea and the compound, for example. And also it's a risky business. So when you multiply all these probabilities of success in the individual stages, you come down to 4% success rate, and this relates to 10,000 compounds that you may need to synthesize to really come up with one drug, or 0 if you are unlucky. So in such case, it's good to stop the project. Here in red is where computations can help. So first, we need to find a hit, a compound that does something to your target, mostly inhibits it or binds at least. And then we can increase its affinity and make it bind stronger, and maybe also optimize some of the properties. As you know, there are 2 general sorts of computer-aided drug design. One is based on the ligand 2D structures that usually comes first, and what you need to know which of the ligands bind or have the effect and which don't. And from there you can go to QSAR, Pharmacophore, and other types. Whereas if you come up with a structure, it used to take some time with X-ray crystallography. Now with AlphaFold you can have a structure. How you trust it, that's another thing. But already you can start with structure-based design. And there the major approaches are docking, which places the ligand in the binding site, and scoring, which tells you what should be its affinity. And these processes enable you to run so-called virtual screening, where you go through huge libraries of compounds and try to fish out those that would bind your target, and you could go further as a clinical candidate, let's say. And also, obviously, calculations are there to save you time and money, not to synthesize and test experimentally all the compounds, but only a limited subset. Now, you can see that also quantum chemistry can be involved in the process. So I advise you to look at this current opinion on structural biology of colleagues from Barcelona who have made recent review of what is happening in the area. Obviously, as you can see, machine learning potentials are there to expedite or make faster the quantum chemical calculations. When we are at the point of predicting affinity, we can go via different pathways. One is based on data or knowledge. and one is based on physics. The knowledge-based, traditionally, the scoring functions, so-called, have been designed in order to be fast. There was the main incentive and objective, and thus there were multiple approximations that had to be taken, which compromised the predictability, let's say. Yeah, and then some parameters, so ad hoc improvements were made in order to have some value. So the accuracy is generally low, but they are fast. Machine learning, obviously, in these days are giving us also quick answers. Okay, the learning procedure may take some time, but still. And the questions there are the input models, so that how much you trust the data you get there, because you need huge quantities of the data. And also the training is done in an area, a domain, where machine learning can interpolate and get you quite interesting results. But going outside of this domain is normally not the task for machine learning. On the other hand, the physics-based methods have traditionally been based on molecular mechanics, which means classical physics applied to molecules. And if you use Newton's equations of motion, then you go to molecular dynamics, where you can get not only movements, but also affinities. I will not speak about free energy perturbation here. I think someone will cover it later, but just for your understanding, this is a method which can use some transformations of like virtual transformations of two compounds in order to get you free energies of the difference in their binding to the protein. And so you don't get delta G of the process where a ligand binds to a protein, but rather you get these two numbers and their difference. So it's delta G of binding. And these methods of free energy perturbation mostly have really made a huge impact and hype, one could say, especially the company Schrödinger with their program FEP+, which is very costly. So they made, they sold it to major pharma companies, and they have used it to quite some success, one could say. But still, such methods are, on one hand, quite hardware intensive. They use GPUs, and they may use days to complete all the calculations. And also, it's still dependent on the force field. So force fields are molecular mechanics parameters, and for some systems and some types of ligands, they may be better. For some worse, what is often the case once it's charged, or change of charge between some ligands, it may become difficult. The other class from physics-based is quantum mechanics. As you can imagine, it will be slow. The current standard for biomolecules is so-called DFT, density functional theory. You would probably run it on tens of CPUs, and it would take you days to optimize the structure to its energy minimum. You can forget about dynamics of the system. So it is relatively accurate. The chemical accuracy, which is 1 kilocalorie per mole, is there mostly. Whether it is applicable to biomolecules, well, you can treat a few hundreds of atoms, so you can, but mostly you would do it in terms of some hybrid schemes like QM, MM, for example, where you link the QM part in around the ligand and the MM, which is further molecular mechanics further around. Okay, why would we want to bother about quantum mechanics when we know it takes time, etc., etc.? So this is exactly because of all the problems that we see with molecular mechanics. Yeah, and these are all the effects which do occur in protein-ligand binding. So I have listed them here, so you can see the non-covalent interactions. That is very generally what holds the ligand in the protein or biomolecular binding site. And these are not only dispersion, you know, van der Waals attraction, hydrogen bonding, which can be either neutral or charged from one side or from both sides, so-called salt bridges, but also some more non-traditional, non-covalent interactions like halogen bonding, which are of completely quantum mechanical origin. We can talk about it later if you wish. So the importance there is that with some level of quantum mechanics, not all, you can reach to a quantitative description. Then metal interactions, they are important. Metalloproteins make up 1/3 of all proteins, so they are clearly important. And you can imagine d orbitals play a role in the binding. So a sphere of 2+, which is normally the description by molecular mechanics, is not enough. Next, polarization, meaning electron density, which can be transferred, or charge transfer, or even hydrogen transfer. These effects do occur in binding and can be described. And also there is covalent binding. So this is a smaller part of drugs, but important drugs like aspirin do bind covalently to their targets, and this is a typical quantum-based effect. Molecular mechanics normally cannot break or form bonds. And as a bonus comes the fact that you don't need to parameterize the ligands for molecular mechanics, because quantum mechanics only needs the elements and charge and multiplicity, for example. So once we said, okay, so let's go for QM, but can we do it? We should consider the timing. So in this graph, where note that the number of atoms on the x-axis is in logarithmic scale, and so in the red curve, the CCSDT is the very precise quantum mechanical method which scales very steeply with system size, and you can maybe calculate tens of atoms on hundreds of CPUs in weeks. Maybe you get slightly better, but you don't really, you cannot make like protein binding sites. Then you have DFT-D. D goes for dispersion, where it's moderate both in terms of hardware requirements and in sizes of the systems. And the last is SQM, semi-empirical quantum mechanical methods, which are very old, like from the 1980s. And their only advantage is that they are fast. So it means they can even go to linear scaling, and but as you can see, the errors of on the order of 10 to 50%. That's worse than molecular mechanics. So what can we do with that? Here we compared to the benchmark level CCSDT, the typical, well, the traditional and one newer SQM method. So you can see the errors have been decreasing over time, let's say over the development, but still over 5%, it's not really great. So the method that Jan Řezáč with Pavel Hobza in Prague took was to try to parameterize the errors. For that, you need some accurate data, and this accurate data came from CCSDT, interaction energies. But as you can not do big systems, they did only dimers of this size, roughly. And they made different parts of it, like for dispersion, for hydrogen bonding, halogen bonding, etc. It's in the NCI Atlas. It now collects 20,000 data points, and it's not only at its minimum, but also some distance scans of these two fragments, angular scans. So all the data have been calculated for the CCSDT and extrapolation to complete basis set. That's details. We can talk about it later. And against this data, the new methods, the SQM methods were compared and systematically corrected. And the results are now in red. So the PM6 is the original method, and D is third generation of dispersion corrections. H is fourth generation of hydrogen bonding corrections. H5 is fifth generation of hydrogen bond corrections. And you can see the errors go down below 5%, which already starts to be interesting for applications. So our working horse is PM6D3H4X or S, depending. So X is for halogen bonding, S is for chalcogen bonding. And such a method can be used. But up to now, we have been only in vacuum. So we obviously need to describe the solvents, and we don't want to treat all individual water molecules and care about their rotations and translations, as is done in molecular dynamics. But instead, we take the implicit solvation model. This means there is a medium which damps the charges. And the traditionally used one was Cosmo. And as you can see, or PCM or SMD, and you can see with this root mean square errors, in some cases it's fine, especially when these compounds are neutral, whereas cations and anions have really troubles. And what Jan Řezáč did was to reparameterize it on bigger data sets, adding the nonpolar solvation term. And you can see in the first line, PM6 Cosmo 2 has even decreased the errors dramatically. So with such a combination of vacuum energies and solvation energies, we could go to SQM scoring. The binding of a ligand in water and protein in water to get the complex can be split in like thinking to these processes. Obviously, in reality, everything happens together and is more complex and is linked, etc. But this is done in order to have clearly defined terms, similar to MMGBSA methods. And the difference is that we use SQM for the interaction and solvation, as we saw. And with such a scoring function, we have designed it in 2010, and since then we have used it in many targets. They are listed here, and in this review it's described. But the trouble is that we have always used one version and then went to another project. So a few years ago we started to ask ourselves, is our scoring-based scoring universal? And to answer this question, we had to really go to the experimental data sets. Because we need to compare to the truth, and the truth is not crystal clear. There was a recent review which is called "The Experiment is the Limit" in Nature Reviews Chemistry, which showed that the distribution of experimental errors in terms of affinities or binding free energies, delta G, is, you can see here, plus minus 2 kilocalories. So, so it's there was an R square of 0.8 in a case where two protein ligand systems were measured in different laboratories. So the reproducibility of the results is not really great. So this is a warning, let's say, for machine learning models where they increase and improve, etc., and they go to correlation of 0.9 with the experiment, but the experiment has its limit at 0.8, so this doesn't really have a meaning. So the data sets, there are multiple data sets out there, but their quality is meant for free energy perturbation, mostly, as I will show you in a while. And there, with dynamics, everything moves and etc. So like the first modeling, like to put like by docking, can be of variable quality, and the method will deal with it. For us, when we use QM, we need a very high quality in terms of structures in the beginning, because we will optimize the structure only, so we will need to be close to the global minimum, hopefully. So that's why we have set out to build a data set ourselves. We named it PL-Rex to remember Tyrannosaurus rex, the dinosaur, but it goes for protein ligand reliable experimental data set. It contains 10 targets. You can see them there. 10 or more ligands per each. High resolution crystal structures, and this means not only the resolution, but also so-called RSCC, real space correlation coefficient, which means that the ligands really sit in the electron density. Also affinities, which were measured in one lab, which should be higher in terms of the reproducibility or reliability, and Ki better than IC50. And also we took care to prepare each protein in terms of protonation, etc., which obviously makes a difference in the scores. If you wish, it's out there in GitHub to be tested. We'd be happy that other scoring methods are tested on this data set. It's challenging. We know it. We built it in this way. So it has large flexible ligands like HIV protease inhibitor. It has halogen bonding as casein kinase with 5 bromine atoms. It has binding via metal like carbonic anhydrase, protonation, deprotonation upon binding, and waters in the binding site. Several waters. We use in this case combination of Cosmo 2 plus several explicit waters. And null hypothesis is we should be better than the fast scoring functions. So as you can see, there are very many scoring functions out there, the standard ones. So we have chosen only the best ones as they came from CASF 2016 data set. And we compared them in terms of R squared. It's on the y-axis, and it means the delta G from experiment correlated to R scores. What you can see when we compare our SQM score with the, in blue, with the standard scoring functions in gray, that we are better. That's reassuring. We are happy about that. But it should be like that, obviously. And what is interesting to see that this red dot means the worst case. And this is an average over 10 targets. So you can see with the standard scoring functions, there are always some targets where the scoring fails, whereas we have consistently high performance. In terms of the machine learning functions, which may not be the newest, they are appearing really quite fast, but we tried to pick good ones. So they don't seem to be very successful. We rationalize this in terms of they have been many times taught on PDB bind, which is a huge collection, but we know there are errors in terms of the structures. So then obviously the machine learning model will learn the errors as well. Interestingly, when we compare the score, this is molecular mechanics and generalized Born implicit solvent, it is pretty bad. But we can also take the geometries from and do our SQM. And we can see it is somewhere intermediate, which means that the success of SQM is for 2 reasons: structures and energies, which is again seen in this graph. Yeah, so I can skip this. When we break it down to individual targets, we can see the consistent performance in the of our SQM 220. We can see the in red are those failed cases, and then when we wanted to go to comparison to DFT as a like quite advanced QM, we had to trim down our system from 2,000 atoms to 1,000. So we recalculated SQM 220 again. It deteriorated slightly, but not very dramatically. And DFT gave pretty similar results, which was for us quite good to see. and obviously the timing was different. Yeah, you can see it here where the accuracy is low for standard and machine learning scoring functions. For us, it's pretty high, similar to DFT, but in terms of timing, we are on the order of minutes, whereas DFT is on the order of hours or days. So now that we've done, let's say, the simple test with standard and more advanced test with DFT, so the real test is with FEP, which runs on GPUs mostly. And okay, we did also some comparison in PLRex, but again, this is a bit biased because it's our data set, so obviously we should be good in our data set. So we wanted to be strict, and so we made a comparison to the Wang data set. This comes from Schrödinger company. It's 2015, a collection of 8 targets. Crystal structures, there were fewer, like at least one per target, but some other ligands have been docked, and so the quality was lower. But never mind, we tried the scoring, and here are, yeah, and also it was, these targets were pretty similar. So this is like by so-called congeneric series, and it is by design meant to make the small changes in the alchemical calculations, whereas we calculate absolute binding free energy, so we don't care how the ligands are similar or not. Never mind. So their result is 0.56 in terms of R square. And you can see 0.23, so CDK2 is failing in their hands, and yeah, 0.42, it may not be great either. And our initial value was 0.38, so we failed in 4 and succeeded in 4. So one could say, well, it's not really great, but as I said, we looked into the structures which have their problems, and these problems we could fix indeed, like 2 hydrogens very close, etc. So we could fix them very easily, manually first, and then with some refined docking, and we could go to 0.47, which is already, yeah, it's worse, but it's, let's say, we are fine with that, and trying to maybe even improve it. Yeah. With AlphaFold 3, that's what I'm doing right now. So now that you have seen like the general workflow, we have some extensions of SQM, and this is this docking. So obviously PLRex has crystal structure, so it's very simple. Once you know the structure already, then it's half work done. Now, how to can we get to the structures other than having the crystal structure? So the traditional approach is docking. The modern one is AlphaFold, as I said. So we started with the traditional one, and we have generated number of conformations, poses with differing RMSD. And now with SQM we are trying to select the correct ones or good ones, or with the best energies, and do the scoring. in this graph you can see the comparison, how we go. So X-ray, it means once we were in the PLRX, so we had X-ray, and the red dot now means that it's like this 20 minutes, like sub-hour per ligand. If we want to replicate this as if we hadn't known the structure, so we do standard docking, we take the best pose, and so the correlation is 0.15, so the correlation is not there. We take 50 poses, and the correlation is better. So already here is a piece of take-home message. Don't take only the best pose from docking. Always use multiple. People normally use 3 or 5, but as you can see, even with 50, we are not really there. So then we made exhaustive docking. So yeah, it needs a lot of calculation. So you can see the red point going up to 1 day, 2 days, and more. We are approaching, let's say, R squared of 0.5 or 6, but it's still costly in terms of calculations. So we are developing this. One other task is native pose identification. That's pretty similar. This we did with 17 protein ligand systems, where you generate a lot of poses, and you want to see which one should be the native pose. In other words, the good binding pose, similar to crystals. So zero-ish RMSD. So you can see in the top right graph that all these points are like energy versus RMSD. And so we take like the curves for 2 systems, acetylcholinesterase and TACE. And this behavior, what you see in the upper one, is quite fine for different scoring functions. In other words, from 0 to 2 angstrom RMSD, you can see 0 relative energy. So that's how it should be, and then it goes up. So this is also fine. All the other poses which are farther in RMSD are worse. But in other cases, like TACE, you can see the points or curves going down in many of the scoring functions, which means that there are some poses which are farther in terms of RMSD, which score better. So this should not happen. So this is the number of false positives. And you can see it in this graph on the right, where you have the individual protein ligand systems on the x-axis, and on the y-axis you have the number of false positives. So in blue you can see in standard scoring functions what you normally can get, so at least tens of or hundreds. And in red, what we used, the 2 variants of SQM scoring, where it was mostly units or 10. And now the last part will be application, and not only protein ligand we can do, but this is protein-protein. And maybe you have seen the poster of Milos yesterday, and if not, probably it is there, so he can tell you a bit more. Also, and it's about a huge system important in diabetes, and that is insulin receptor plus insulin. So IR is a huge protein, transmembrane, you can see it here. In the outer part, is shown like in the domains L1, CR, L2, etc. And you can see in green and black helices, that is the insulin sitting on it or binding to it. The available structure data are low quality. We cannot choose. It's you can see Nature papers or Science papers from 2006. So it's like every few years some high profile journal and some special structure, but the resolutions have never gone below 3 angstroms. So we must imply some modeling. We have the insulin, and it's 51 amino acids. It's some of them are exchanged in analogs, which are used then for diabetes treatment. So it's pretty important to understand this computationally. And also, this is one of the cases where you can see the continuum of conformations of insulin receptor. So we should think about molecular dynamics. Yeah? We don't want to optimize something that we even don't know. So that's what we did. We ran molecular dynamics. But in the same time as we want to use quantum mechanics afterwards, we will need to like select some part of it. We need to make sure that some movements are there, but not gross conformational rearrangements, because we couldn't then compare. So this is a case where you may see in sticks this is cryo-EM structure, and in lines there are several snapshots, so you can see how the movements are. So, and even you can see the loops that do move enough, I would say. And also you can see, like from 3 trajectories that we ran, some percentages of hydrogen bonding between pairs of insulin residues and insulin receptor residues. So you can see in green some of those that hold very, very tight, whereas some of others in yellow or red are very flexible, are there in some trajectories and not in others, or are not there at all, etc. So what to learn from this? We wanted to quantify how individual residues contribute to the binding. So here are like this B13, A4, B16 are amino acids from the insulin, where we did so-called virtual glycine scan. What does that mean? That means that we calculate the interaction of the insulin with insulin receptor, and then we exchange the residue in question. So let's say this B13 glutamate with glycine, and calculate the interaction again. And the difference between these two is the contribution of this side chain. Yeah, so these columns mean how much these side chains contribute to the binding. Obviously, you see this 30, 20, 12. It's huge, these energies, and it's connected with. You can also see that you have arginine plus, glutamate minus arginine plus. So these like highly charged areas are very difficult even for quantum chemistry. So this red and yellow are 2 sorts of SQM. So you can see even 10 kilocalories difference between them. The other part of variability are the error bars, which are rather like differences from 10 conformation, that's averages and standard deviations. And on the right-hand side, you can see that the different methods give roughly similar results with differences of a few kilocalories, but still going from the right to the left, from molecular mechanics to SQM, which like should give more reliable data. Down here in violet, you can see how these SQM energies differ from 3 trajectories. So also a few kilocalories difference can be seen. All right, so I think I should stop here by summing up that with SQM 220 we have a universal, up to now, as we can say, physics-based quantum mechanical scoring, which gives DFT accuracy predictions within 20 minutes on one CPU. The SQM geometries, which are also part of the PLREX data set to be downloaded, can give you really information on how these binding modes, what non-covalent interactions are important. And we have now tested it on other data sets like this FEP Wang data set. Yeah, so we are also having clients from pharmaceutical companies. We have one US, one European, which buy this software to use it, and we are trying to develop it. Also, we have some like machine learning corrections now. So I think the direction of SQM will thrive and will continue to be useful in the drug design. All right, so that's all for me. I should acknowledge Pavel Hobza, who started these ideas and this work on SQM and quantum mechanical methods decades ago, and we were part of his team. Before then, HPCG team of Petro is here, and this is high performance computing at our institute, which really gives great support in terms of the supercomputer and calculations. IOCB Tech in terms of the licensing, and Gachar also for this insulin part. And that's all for me. Thank you for your attention, and if you have questions, I'm ready to answer. And if not, I'm not sure who's the next one. Okay, we may check the program.
Thank you, Carol. So I think this works. So basically, as Carol said, I'm working in different places, and I'm kind of mixing work in academia and in industry. And I will try to give you kind of perspective for artificial intelligence and drug discovery, mostly from industrial and business point of view, because I'm involved in the company which is developing AI tools for drug discovery. So first, I want to start with kind of uncomfortable truths about drug discovery in general, because when you enter the field, you mostly consider it to be like this, like highly automated pipeline, where everything is shiny and automatic, but the truth, the reality is more like this. So there is a lot of manual work still, despite all the technological progress which we have now. And that's one of the reasons why drug discovery actually struggles nowadays. So the plan of the talk is more or less like this. First, we will try to discuss why it struggles, what we can do to make it struggle a bit less. And after that, I will show some cases from our practice, how we actually use AI to solve these kind of problems. First of all, the reasons of widely recognized stagnation in the drug discovery is that the cost of drug development actually increases and the development time doesn't change that much. So the last survey in 2022 tells that only about 6% of all drugs which enter the pipeline are successful. So the composite success rate is about 6.3%. And as a result, the failure rate is more than 90%. And this figure doesn't change last like 20, 30 years. And as you can see here, there is a funnel of drug discovery where you go from the target validation to the clinical trials, and on each stage you are losing some of the candidates unless you end up with this 6%. Another thing which suggests that we are actually kind of cursed is called the Erum's law. And Erum is not a person, it's just a Moore which is pronounced backwards. And all of you know Moore's law, that there is an exponential growth of the computer power. And Erum's law is the opposite, it's exponential slowing down of drug discovery. And here you can see that the cost per drug actually increases exponentially. This is logarithmic coordinates. And up to 2010, you get a figure about 1 billion for less than one drug. So one drug costs more than $1 billion on average. And this is a lot of money, and it's only getting worse. So actually, the explanation of this Erum's law is quite complicated and not really clear why it's like this, but widely recognized that the first problem is that it's very hard to beat established treatments. So you need to invest more and more to get something which is economically viable on the market. Then the regulators are going insane more and more, so there is a burden of clinical trials which is increasing. And there is a tendency that more and more money is allocated for R&D, but you have a problem that one woman cannot give birth 9 women cannot give birth in 1 month. Yeah, so if you invest a lot of money, you cannot speed it up more than it naturally can be. And also it's a basic research bias, which means that we are kind of overestimating the value of a basic research. We are throwing more and more money in this, we are collecting a lot of data, but the output is not proportional to this. And one of the things how we can beat this Erum law is actually this AI, because AI is now considered kind of game changer. The paradigm change, and we estimate that if everything will go as it is supposed to be with AI adoption, that the cost of the drug will drop by 60% by 2030. This is an optimistic estimate, of course. And there are some ideas how to deal with all these problems, how to cut the R&D cost, how to deal with regulators and all this stuff. And what I like the most, that throw the money at the research, but let's better throw the money at AI. This works very well right now, so we all see that in America they allocate like 50 billion for AI studies for the next years, and not most, but some of this money will go to the drug discovery as well. And as you can see, if all this is true, then probably we will get out of this Erum's exponential law, and we will save quite a lot, more than 1 billion by year 2030 on one drug on the market. So which problems actually AI can solve? It's quite a lot of them, and I kind of classified them in 3 categories. The first category is the problem of the context gap. So when you're doing drug discovery, you're working in multiple different domains. You need to know chemistry, biology, simulations, bioinformatics, different omics, population omics, patient data. All this is very different. So in your team, you need to have like 5 to 10 people which are experts in these fields, and after that you need to connect them together somehow. You have to manage all this. It becomes a nightmare very quickly. And with AI, you can kind of bridge these gaps, so you can have kind of explanatory model which accumulates all the knowledge and explains to the person who is not an expert what's going on in the next field. The next problem is intractable amount of data. So the modern drug discovery is all about data, and the amounts are really quite large. So we are routinely dealing with chemical spaces of about 50 to 100 billion compounds. And now some of the vendors are providing trillions of compounds as a combinatorial databases. It's insane amount, not human comprehensible. Then if we are speaking about pharmacokinetics and pharmacodynamics, for each of the compounds, you are usually evaluating from 30 to 50 parameters, which is also quickly becoming quite tedious to deal with. Then you have a lot of high throughput techniques. You have HTS, you have Dell screening, RNA displays, phage displays, you name it. And this produces huge amounts of data. You have to deal with this somehow. Then you have trial outcomes, also lots of data for many patients with all the endpoints, biomarkers, whatever. So this intractable amount of data have to be digested and somehow processed. And AI can help with this. And the last but not the least category is workflow construction. So you have a lot of different techniques, in silico experimental techniques, in vitro model, animal models. Which of them you should use for this particular problem on this particular stage of the drug discovery is not actually so obvious. And the building the pipeline is crucial because if you choose the method incorrectly, you will most likely fail and you will lose money and lose time. So with AI workflow construction, this actually allows you to choose the optimal strategy based on the previous experience and some heuristics which you have from the data. So in a way, in the traditional approach, you are forced to choose between doing this quickly, reliably, and cheaply, and you can only choose two of them. And with AI, the bright promise that you can do this all at once. It doesn't work like this yet, but there is a hope. So in terms of concrete applications, we can divide this into the stages. So when we start with target identification, this is the very first stage of drug discovery. You can use AI in multi-omics to combine the results from genomics, transcriptomics, proteomics, interactomics, lipidomics, whatever omics. Then you can construct knowledge graphs, kind of databases where you have the data from many different domains and relations between them. Doing this by hand is almost impossible, so with AI you can fill this graph and after that use it in your research. And of course you can do unstructured data scraping. You can use LLMs for reading papers, patents, clinical studies, reports to extract the information you need. Then when we go to early discovery, we have several points here. First, you can use AI for de novo molecular generation. You can use it for AI virtual screening of existing chemical databases. You can use it for ADMET prediction to predict multiple parameters of pharmacokinetics and pharmacodynamics. You can try to use it for automatic user comprehension. So when you have a lot of compounds and you don't quite understand which modifications of the compounds lead to the outcome you see, you can use AI to explain you what's going on the chemical point of view. Then you can also use it for drug repurposing and for some other things. So once you get the molecule, which goes to the late stage of the discovery, you can also use AI, for example, to formulate formulations of drug delivery. You can prepare for IND in clinical studies. You can predict the outcomes of these and decide which molecules should go there and which should not. And of course, you can do all the kind of data mining for patient clearance and so on. One thing which is also kind of gaining momentum now is simulated in vivo testing and clinical studies. And coming to the clinical studies, again, you can use AI for forming studying plan and monitoring the clinical studies, for predicting risk factors, for automating patient recruitment, and so on. And again, last but not least, is data management. So all these stages are producing a lot of data, so you can automate data mining and integration. You can do quality assessment and with AI and so on and so forth. So let's dive a little bit deeper into AI in the early drug discovery. The first thing, which all of you probably know, is protein structure prediction. So AlphaFold is probably the most valuable AI model of the last 10 years. And as I know, Carl has a whole lecture about this. I don't know if it was yesterday or not, but anyway, so I would not go too deep into this. AlphaFold is not only the model, there is also RosettaFold and some other models which are doing the same. Then with AI, you can do binding pocket prediction. When you don't know where your molecule binds on the protein, you can try to predict it with AI techniques. Then you can generate chemical spaces. You can predict the ligand pose in the binding pocket, which is often called AI docking, which works like, the outcome is like conventional docking, but the methodology is absolutely different. And then you can predict different dynamic properties. You can generate protein ensembles with AI, or you can try to find transient and cryptic binding pockets. And I want to showcase how you can use AI for binding pocket prioritization. This is a work which we did this last year in the company. And this is fancy because we try to use LLMs for this binding pocket prioritization. And usually it's funny when I mention LLM, everybody in the audience is actually awaken and people are no more sleeping. So LLM is kind of being seen now, everybody is speaking about it, but how good it is in this kind of real world situation. So the problem is that if you don't know where your compound binds, you can find kind of pocket-like cavities on the protein surface, which are kind of visualized here. And after that, you should somehow select which of these cavities are real binding pockets and do the virtual screening against these. There are a lot of algorithmic techniques of doing this, like tens of them. One of them, which is arguably the most usable and reliable, is F-pocket. And this actually gives you kind of heat map on the protein surface where the binding pockets could be located. The problem is that it predicts much more pocket-like cavities than are actually biologically justified. So after this stage, you have to go into the literature and you should manually prioritize the pockets. You should find which residues are really involved in the ligand binding according to experiments like mutational studies or interaction with other proteins or interaction with some fragments or whatever. And this is very tedious, so you have to do it by hand. It's not a problem if you're doing PhD, for example, you're working with one protein, but in the industry scale, let's say last year, we had to did it more than 40 times in the company, and it's become problematic. So we want to automate it, and the question is, can we use LLMs to go into the literature and to extract information we need? So we did kind of computational experiments. We took 10 proteins from very different protein families, which are listed here. So there are all kinds of proteins, water-soluble, membrane proteins, enzymes, whatever. So it's really very wide variety of protein classes. And for each of these proteins, we first extracted 3D structures from PDB, and we also found high-quality peer-reviewed articles, about from 4 to 7 articles per protein. And these articles were classified into several categories. So the easiest rank is when this article describes just one binding pocket for one protein, the easiest way. Then the articles which describe multiple pockets for the same protein, then multiple pockets for multiple proteins, which is quite hard to parse, and the negative control where no pocket information at all is in this article. After that, we did the baseline testing with human experts. So we asked several human experts to go through all these articles and to annotate the binding pockets which are described there. And after that, we were comparing the LLM results with this baseline. The LLM query is kind of like this. So we are asking the LLM to consider that it is a senior medicinal chemist, to condition it to the biological domain, and we ask it to determine the number of binding pockets for the target protein described in the paper, to make very specific discriminating characteristics of each of the pockets, and to give the list of amino acids for each of the pockets. And after comparison with a human baseline, we get the results like this. So we started this work at the time of GPT 3.5. You can see that it was failing most of the time. After that, when GPT-4 was out, the results improved to about 60%. And you can improve it a little bit more if you fine-tune this kind of prompt by adding clarification like, do not treat numbers which are not residues as residues. Because the model is stupid enough that if you see the number like A25, it can actually decide that this is alanine 25, when this is a number, this is a strain number of bacteria or whatever. So in general, we can get success up to 75%, 76% with GPT-4. But it appears that this is not end of the story. When GPT-4o was out, we retested everything, and the results was a little bit better. But what is interesting is that if you take the GPT-O mini, which is kind of smaller and much cheaper model, the results are exactly the same, as you can see here. So you can pay no attention to this chunk size. I will not dive into it, but the results are comparable for the large model and for the small model. So you can pay much less and get the same results, which is interesting. Of course, it's not all roses, because LLM is very good in filtering out the bad binding pockets, but it's not so good in combining them together. So for example, in many cases, it identifies several pockets where they actually constitute one large pocket, like on this figure. So there is a need for post-processing to merge the pockets together and to tidy them up a little bit, but nevertheless, it works quite good. So the conclusion is that the accuracy of LLM prioritization with the latest GPT-4o is overall quite decent, but it's not great. It's still not on par with humans, and you still need various tricks for post-processing. But the LLMs are progressing very fast from one hand. From the other hand, they are becoming very expensive. So for example, the next reasoning O1 model is much more expensive in terms of the API, and now we are waiting for O3, and we don't know how much it will cost. Maybe it would be more, it would be better to rely on humans on this, depending on the cost. But anyway, this is promising, and in the next decade, we will see more and more such things with processing of the literature information with LLMs. Okay, another thing which I want to go to is AI virtual screening. So when you know where your molecule should bind, you should select the molecule itself. And this is a kind of very complicated slide for our business presentation, where we kind of showcase all the places where AI is applied in the virtual screening. I will not go too much into the detail here, but here what we can do, we are doing the screening in 2 stages. And on the first stage, we are using AI to do the rough screening of the large chemical space. And this is done with kind of semi-ligand-based AI model, which is trained on the combination of the protein and the ligand without knowing any structural information. It just knows that this ligand binds to this protein, and it has some quantitative estimate of affinity of binding. And the model trained on this can actually work very fast, and it can go through millions of compounds in a minute. And by doing this, we can quickly go through large chemical spaces and prioritize the molecules. After that, we have dedicated AI models for selectivity on the proteome-wide level. Again, we are taking single compound, and we are trying to match it with all the proteins from the human proteome, about 17,000, which we have right now. And if this compound binds to many of them, that this compound is not selective, so we're getting rid of this compound. Then you can also predict ADME and toxicity parameters with yet another AI model. I will come to this a little bit later. And finally, you can do AI docking, the final screening, the final scene. And this AI docking is actually substitution of the conventional physics-based docking, so it provides the same result. It provides you the binding pose of the compound. And the good thing about all this AI approach is that you can collect all the experimental validation data. After we provide the compounds to the labs, they do all the validations, they provide the data back, and we can use this data to fine-tune the models. So on the next stage, the models already understand which part of the chemical space is active and which one is not. And it will no longer go to the inactive chemical space. So usually the success rate increases quite a lot on each next iteration. And empirically, we know that usually on the optimization stage, where humans need like 10 iterations, this AI technique can do the same in like 3 iterations. So it's quite a lot of gain in time and cost. So I will skip this one. So coming to ADMET prediction. So when we're speaking about pharmacokinetics and pharmacodynamics, we have currently an AI model which is predicting all these parameters, which are listed on these slides, at once for each of the compounds. This is a multitask AI model. So instead of training the dedicated model for each of the endpoints, we are training the model which is predicting the vector of endpoints, all of them at once. It works much faster, and it also has an advantage that it's kind of cross-disseminating from different endpoints. If you miss the data from one of them, the model can use the data from the similar compounds for another endpoint. And here is a case study how it actually looks like in one particular endpoint, which is a membrane permeability of the compounds. So we did it in collaboration with Carlo here in University of Olomouc. And actually, we took the data from MolMeDB, which is a database of permeability, which is developed here. And we used our multitask ADMET technique, our neural network architecture, to predict this. And immediately we see that the numbers are quite suspicious here. So 0.97, it means that we have 97% accuracy in predicting the permeability. And this is really suspicious. This is too good to be true. Because all the other models in literature are less than this by a large margin. So of course we are smart, but we don't believe that this is true. So we tried to understand what's going on. And we immediately found that there is a problem in the data. So most of the time, if you see an artifact like this, it's either overfitting, which is not the case, or it's a problem in the data. So actually what was available in the MolMeDB appears to be not the raw data, but the predicted data. So it was very ancient from 2002 neural network model, which was predicting permeability, and the data in the database were actually predictions. And this is the same story when you are using AI, which is trained on AI-generated text. It degrades. It's the same story here. If you're using AI on AI-predicted values, you are getting something insane. It's close to 100%, but it's not true. The problem was that the real experimental data was no longer available. So when we did it in 2020, all the links are already broken, and it was claimed that the data set was actually moved to the US Environmental Protection Agency, the governmental institution, but on the side of this institution, all the links are also broken. So we were kind of at the dead end. There is no experimental data, so we had to do some data archaeology. So we went into the paper where they used this initial FISPROB database, which is no longer available, to curate it, and after that they published a curated subset of this database, which is, thanks God, available up to date. So we use this, and you can see that even in the curated data set, there are some invalid compounds. So the curation was not that good. And after all of these, about 14,000 compounds were made from initial like 15,000. So we use this to retrain the model, and this is a message to US Environmental Agency, good job. And this is how it looks like after the retraining the model on the real data. So from 97, we are back to 94, which is still okay, but it's not insane. So this is in line with the previous data, which was about 93%. And here I want to show you how predictions of our multitask AI model in terms of ADMET parameters compare with other models which are available. There is a TDC open benchmark. There are 22 ADMET endpoints. This is a public benchmark, public leaderboard. And we are overall the best on this leaderboard right now, and we want to participate officially this year. Then we are coming to AI docking. So AI docking is an interesting beast because this is not docking, and it's not exactly AI. So it's like the machine learning model for something which produces the same output as docking. So the models for this are usually trained on the existing protein ligand complexes. And after that, you provide either the smiles of the compound or the 3D conformer of the compound, and the model predicts how these compounds lie in the binding pockets. The problem is the data, again, because there is a very limited number of experimentally determined protein ligand complexes. There are about 55,000 of such complexes altogether. If you want to concentrate on the complexes where binding affinity is measured, it's even less, it's less than 20,000. And if you want the high quality binding affinity, it's less than 10,000. And it's also very much skewed data set because, for example, for more than 1,500 ligands, you have only one complex. So the ligand is only crystallized with what is one protein. There is no variety. About 1,500 proteins are constituting 80% of all the ligands. And there are 100 protein families which dominate the data set, while the total number of protein families is much more than this. So this is very limited and very skewed data set for machine learning. And in order to overcome this somehow, we are using data augmentation techniques. So the idea here is to take the statistical distribution of interaction in the real protein ligand complexes and generate kind of artificial binding pockets around other ligands in such a way that these complexes follow the same statistical distributions. So by doing this, you can generate as many as you want. You can do millions of such complexes, and you can present them to the model. And the assumption here is that all the major interactions between the proteins and ligands are already present in the experimental data on PDB, but there are not enough combinations. And by doing these artificial complexes, we are kind of sampling all these combinatorial variants and providing these combinations to the model. And this is how it looks like. This is figures from our paper. So for example, you can see how different kind of hydrophobic interactions look like in terms of the distribution from experiment and from artificial pockets. They're not exactly the same, but the trend is there, and this is enough for model to understand how it works. And the same for H bonds, for example. Again, this is not ideal, but this should not be ideal. It should be roughly the same trend, which is there. So there is a reasonable correspondence, and after applying this data to the model, we are getting quite good results. And we use this in our RT-doc AI docking model, which is the best model currently on the market. So I will show you the benchmarks. So the distinctive features of this model is quite small. Because if you take AlphaFold, for example, AlphaFold is kind of monstrous AI model. The architecture is extremely complex. It's one of the most complex models out there. And our model is quite light. And we are actually compensating for this, but throwing in more data to train it. It also includes only the binding pocket to make it even lighter. And as I said, it is augmented with these synthetic protein ligand complexes. And it's also able to integrate the protein dynamics. Because since the model is very fast and very light, we can do ensemble of conformation instead of one conformation. And we can also train the model on the trajectories from MD simulations. So this is how it looks like in terms of the benchmarking. This is very well-known Aztecs data set, which is already considered outdated, but still widely used. It's not particularly useful for assessing AI models, but you can see that the AI model is actually much better than the conventional docking models and other AI techniques. But there is another data set which was actually created to challenge AI models, which is called Postbusters data set. This was introduced like 3 years ago and includes multiple structures which are challenging for AI techniques back then. And it also includes multiple structural quality metrics beyond just the MSD of the pose with the crystal structure. And this is very, very valuable because before Postbusters, it was no clear distinction between the molecules with the same MSD, but kind of distorted chemistry. So this is how it looks like. So here on the right, you can see different previous AI models, and you can also see AlphaFold latest model, which also can predict the protein ligand complexes, and different variation of our RT-doc model. And this is the last two of them, 1.8 and 2.0. The difference is that 1.8 is actually trained to get the absolute best result for the RMSD, but not necessarily with these additional structural metrics. And 2.0 is actually minimizing the gap between these. So it produces overall the best results. And if you will compare with the conventional docking, you see that we have an edge about 20% in terms of accuracy. And the gap here, the shaded area here, is as small as for conventional docking. For other AI techniques, as you can see, it's quite large. So another thing about RTDoc is the speed. So when you compare it with AlphaFold, for example, AlphaFold is better than RTDoc in terms of the overall accuracy, but it's also 600 times slower. So when we're speaking about virtual screening, it's not possible to use AlphaFold 3 or any AlphaFold-like architecture for virtual screening. With RTDoc, we are only taking about 1 second per one compound, so we can use it in virtual screening quite easily. And it's surprisingly, it's also better than the conventional docking. So if you're using Vina, for example, which you will use here on the hands-on session, Vina takes about 3 minutes on the same hardware, where RTDoc takes about 1 second. And this is an advantage of AI techniques in general. They could be much faster. There are also some detailed comparison with other things like glide or unimode docking. I would not probably stop on this too much, and I will jump directly to the conclusions. So the first thing we all need to understand that AI drug discovery techniques are here to stay for a long time, and most likely in the next decade, we will see that this is the decade of AI in all drug discovery fields. The pharma companies are quite slow in adoption, but it increases. And of course, we will see more and more usage of LLMs for data mining and analysis of the literature data, also in writing the papers, which is another problem which I don't want to discuss here. We will also probably see progressive substitution of the physics-based techniques by data-driven techniques. So maybe finally the conventional docking will die, we will see. And of course, data is new oil, so we need the data to feed all these AI models, but nobody wants to collect and curate this data. So if you feel like you can produce the data and compile the data set and make it online, this is probably the best way to boost your citation index. And thank you for your attention. So, any questions?
I was wondering, like, how you implemented ChatGPT 4.0 for the pocket mining, and specifically, what level of prompt engineering you employed? It was like a little bit of it, or it was a very deep JSON-based with dictionary to map the residues, and then to counter map with fpocket that ChatGPT can call fpocket and so on. What level?
So here LLM was only used to get the list of residues from the literature, and after that all this story about backmapping and combining together and merging, all this is algorithmic. Because we tried to do this with LLM, it's not as good in this. So you should really use kind of a reasoning model to really explain it, what it should do, and the reasoning models are extremely expensive, because all this is done with API calls. So you are paying a few cents per token, and this is quite expensive. That's why it was important for us to see that the mini model is actually performing as good as a large model. But of course, the prompt engineering is a bottleneck here. So you don't really understand which kind of prompt you need to produce. So you see that after some light tuning, you are improving the results. But this was the maximum which we were able to achieve. Of course, we can fiddle with this a little bit more, but pretty much the prompt was like this. So it's not very elaborate.
Another thing that I'm thinking about, and somehow, can you imagine to embed the mining operation that this kind of model are able to do? Not the reasoning one, because it's too expensive and not worth it probably, but the standard one, stupid one, let's say. Are anyway very good in mining information about stuff, and if you code it in a way or in the other a little bit, you can feed them into classical machine learning or classical deep learning framework like the one you shared later on with Art. Have you
so this is actually doable, and we actually do this. I didn't have the slides here, so it was about, where it was, let me see, the knowledge graphs. Actually, for these knowledge graphs, the knowledge graph itself is just a graph database. So you have some entities, and you have relations between them. The relations could be something inside something, or something be associated with something else, or you may have something specific like this is a target for this drug, or this is a ligand for this protein. But in order to construct this kind of graph database, you need somehow to scrape all the data from the structured sources and from the literature. And here, this is exactly what you're asking about. So here, the LLMs actually shine, because you can extract the data. If it produces a certain number of false positives or false negatives, it's acceptable. So once you have the graph, you can reweight it, and if you have the uncertainty in the weight of the edge on the graph below, let's say 0.3, you can just delete this edge, and you are getting rid of the most of the false positives. So this is how we are doing it right now.
Was about multitask prediction and your results about log-p prediction. You said that your prediction was 0.94 and the previous model was 0.93.
93, yeah.
Do you remember the previous model? It was single task model, yes?
It was single task.
Do you remember what it was the machine learning methods?
I don't remember which architecture was there, but I mean, all these models are kind of the same, because you are getting molecular descriptors of the molecules, you encode them, and after that you are just matching with a measured permeability. Yeah.
What is your idea why the results of your model and previous one is so close? Maybe it's because they had enough data.
Yes, absolutely.
The results is very high, already very high.
This is a natural limit for the data set which is available. So if we will, that's why I say that when we get 97, we immediately understand that something is wrong. Because there is a ceiling which is set by the quality of your data. If you are getting 99, you are overfitting, for sure. If you are getting 97, it's not exactly overfitting, but something is wrong. But the results are quite good anyway.
So actually, in this case, we need to expand the applicability domain for the model and add new data from with other compounds maybe, and then we will get another results maybe better.
Yeah.
So it's also related with one of your conclusions, the last one, that you told that the data is our oil for researchers, but actually it was for years, for the decades.
Sure.
And you told about AI in drug discovery. What are you thinking about if AI will get data from articles? Because there are a lot of projects who try to apply AI to
Retrieve the data, yeah.
data from articles. What do you think about it? I think we will get it in the next years. But what you are thinking about?
Well, you see, it's actually the same as the previous question. So we are using it right now already, but it's kind of limited in kind of what information you can retrieve. So, for example, you can work reasonably well with a text. You cannot work with figures. And you're working really bad with tables, depending on how it's set up in the PDF file. So you have to use some OCR or machine vision to actually make a photo of your page, and after that to understand where the table is and what are the values. It's a mess, I would say. So right now, the scraping of the scientific literature is a problem which is not solved yet. There are several companies which are doing dedicated exactly this, trying to go through the PDFs and extract as much information as possible. But for the scientific literature, it's not solved. So maybe in 5 years, we will be there.
I do remember that when the div doc is used, then you can quite often came up with the unphysical close distances of the atoms of the ligand towards the protein. How have you overcome this type of problem when the structure is generated within the protein structure?
Yeah, basically it's here. I didn't speak about this too much, so let me find it. So this plot is kind of all the metrics of the chemical quality metrics, which are in the Postbusters data set. And this is a minimal protein ligand distance. And you can see immediately that, for example, for DivDoc, which is gray here, it actually fails. So it's about 30% only of correct predictions. And this RTDoc, we are about 75, and this AlphaFold, it's about 80. So it's not 100, even with the AlphaFold. And the way how we did it, first, when you train your model, you can actually pay more attention to this. So you can increase the weight and tell the model, if it bumps, please punish this structure. Another way, you're always doing post-processing. So what these models predict, they predict in the matrix of the contacts. And you have to translate it somehow to the coordinates. And in the AlphaFold, they are using in AlphaFold 2, it was kind of complicated procedure. In AlphaFold 3, they have a dedicated diffusion model to do this, which is different. The results are roughly the same, by the way, and the speed is roughly the same. For us, we are first using a differential evolution algorithm. So this is an algorithmic approach when you have the matrix of contacts and you convert it to the point cloud. And after that, on this point cloud, you are doing post-processing. So you have to use some physics-based techniques to get rid of this. And actually, if I will go here, so the difference between this and this is actually in the usage of the post-processing. So without post-processing, you are getting very high RMSD value, but not as high chemical correctness. If you are applying post-processing and you are physically moving the atoms a little bit to minimize the clashes, you are actually decreasing the top one, but you are increasing the bottom one, and you are making them converge together. And this is the same what the conventional docking do, because conventional docking is using physics-based from the very beginning. Yeah. Any other questions? Okay, it seems that we are done.
So now we have 1 hour pause, a long pause for the lunch. There is possibility to go to lunch upstairs, or there is the baguettes downstairs, and I think that
Test, test, test. Wonderful.
okay. Welcome back after the lunch pause, and we will be continuing with the Pharmacophores, which will be
I guess, yes. Yes, I love it. So, thank you very much for this kind introduction, Karel, and thank you for inviting me again and again and again. I really love to come here. Unfortunately, the time of the year is not ideal because we are very busy in this period, because in January we still have tons of students and the semester break starts next week. So I need to get again back to Vienna very soon, so I cannot stay here, which I'm really sorry about. So today I will give you this introduction here to Pharmacophores, but more I will talk about a little bit about what we are doing for next generation pharmacophore modeling. So it's not the basics actually, and later on we will have a tutorial which I really love to do interactively with you guys. So we will move up to the next room and then we will play. And I enjoy this, and I hope that you will enjoy too. So first of all, before I forget at the end, I want to acknowledge all my people here. This is a snapshot from last year. Actually, it's changed already. A few people are no longer here. A few more joined, but this is a snapshot of my group in Vienna, and I'm in Vienna since 11 years now. I just want to highlight this guy here. This is a senior postdoc, Thomas Seidel, who will be mentioned later on. This is a postdoc here, Oliver Wieder. This is a postdoc here, Verena Battisti. This is a postdoc, Christian Perman. The other one are PhD students, Christian Fellinger, Daniel Rose, for instance, and interns, and Sarah Tkaczyk from Poland. And the other ones are also Sad Nedra is from Italy here, and the other ones are interns coming and going, and so there's quite a dynamics here. But before I came to Vienna, I did some other things, and actually I spent a few years in France in industry. Before that, I was in Innsbruck, and I had created a company called Intelligent 21 years ago. And so I have to acknowledge a lot of people here working together with me in Innsbruck, in the company, and here these are the people that are now in Vienna, mostly contributing to the modern things that we are doing. And of course, I have also to acknowledge some funding. Without funding, this would not be possible. So before we go into the more details, I just want to mention here one very, very important thing. Whenever you do computation, simulation, in silico, whatever kind of experiment, you need to validate them. It makes no sense to make a model and then that's it, right? You need to use this model and do some experiments to validate it or devalidate it, right? And the model doesn't need to be right as long as the model gives the right answer, right? The model does not need to be correct unless we talk about physical models, then they need to be correct. But if you have a model that predicts something and the prediction is true at the end, we don't care if the model was right or wrong from the algorithmic point of view. The result must be okay. And so we have this drug make, design, make, analyze, and test. This cycle here in rational drug design, where we try here to develop methods that are in this area here, the rational design, so that we can try to not waste too much time and efforts in making compounds that will not make it at the end. So compounds that might be not efficient enough, might be toxic, might have some other undesirable problems, so that right from the beginning on we can make a hierarchical ranking of compounds, I would say. But again, only experiments will validate your models. And when you think about ligand binding, actually this is really something, understanding ligand binding is one of the most important things to the target, of course, but also to potential anti-targets. And you had a very good lecture this morning by Martin Lepšík, of course, who I know very well, and he's fantastic here, does groundbreaking work. But we need to answer these questions, early drug discovery, especially when it goes to hit identification, hit to lead expansion, and lead optimization. And it's always like finding a needle in a haystack, right? And I will get back to this picture a few times during this lecture, you will see, because we believe that you need really good methods to find the needle in this big haystack. And now the trend is to make the haystack even bigger, because there's so many compounds that can be done and are already in the virtual space. So how can we do this? So it's about data analysis, which molecules to choose for biological assessment, which one to make, which one, how to modify the molecules for optimizing the properties. You know, we have a multidimensional optimization problem when we do drug discovery. We need compounds that are not toxic, that are well soluble, that have a high potency, a high efficiency, et cetera, et cetera. There's hundreds of parameters that we need to optimize. And we need models for helping the medicinal chemists to make good decisions. Also avoid risks of toxicity. And also we need methods to represent our data in a way that medicinal chemists can make sense of it. So how to guide analysis of molecular modeling results, and how to make use, for instance, of MD simulations. MD simulations are things that are coming on more and more because computer resources are becoming more available. And they are now even in industry. So it's not only academic exercise, but it's also in industry that people do more and more MD simulations. But then there's a ton of data that is generated. How do you represent this data? How do you use it to make the right decisions? So a possible way to do this kind of analysis is to compare compounds with each other in the view of their preferences for undergoing specific molecular interactions. Of course, it's always about molecules interacting with each other. Small molecules with big molecules, small molecules, drugs with proteins, for instance. So what you can do is you can look at the structure of a small organic molecule and annotate these structures with all potential interaction features that such a molecule could do in theory. And for this, you can use pharmacophore descriptions, and here you have a picture here in the way how we do it. So this would be all the features that this molecule could do in theory. But does this molecule really do all the interaction in its target? So we need to find out which of them are the real important interactions, right? Maybe not all of them are important. So we can do this considering only those interactions of the ligand that are visible, for instance, in the protein binding site. And this is very easy. If we have an X-ray structure of a ligand co-crystallized, then we can, with a click of a button, generate such a pharmacophore model that shows only the interactions that this ligand makes with the amino acid, the corresponding one, in the protein. So for instance, there was an H bond here at this keto group that could theoretically accept 2 hydrogen bonds, 2 lone pairs, but there is no here in the protein because there is no H bond donors opposite of this keto group. So we can learn that this is probably not such important to have this there, right? And this is true for the other features that we don't observe here. So we learn from X-ray structures which are the features that are important. So a pharmacophore, this is the definition, the official definition of what a pharmacophore is. This has been coined by Camille Vermouth. He was a very famous medicinal chemist. He worked his entire life in academia, but he discovered 3 drugs that made it to the market, together with industry, of course. And he was president of the UPAC section for medicinal chemistry for many years. And this was the term that is the official term of the UPAC, what a pharmacophore is. Sometimes you read pharmacophore descriptions in books that are not correct. You cannot say, for instance, a guanidino group is a pharmacophore. This is not true. The pharmacophore is the entire description of the interactions of a molecule with another one. And so these chemical features that represent the defined binding mode of a ligand to its biomolecular target are mainly electrostatic interactions, H bonding, aromatic interactions, hydrophobic interactions, coordination to metal ions, also halogen bond, for instance. And this is a language that is very easy to understand. This complex way of molecules interacting with each other transformed into a meta-language that is representable in 3D, in 2D, as you see here. And so this is a language that helps people to communicate with each other on one side, but also to use this for designing molecules, to do virtual screening, for instance, or overlaying molecules with each other and find out which common features are between molecules. So I believe still, and I will not stop believing this, that pharmacophore models are the most suitable data representation methods for guiding medicinal chemistry efforts, because medicinal chemists think already in pharmacophores, and if they see them, they're even more comfortable. And you can discuss what kind of groups you can exchange but keep the same interaction, or making a specific interaction even stronger, for instance. So you might think that it's not a new story, right? Pharmacophores, it might be seen as an old-fashioned concept. Of course, I mean, there is even a guy who said very early on in the beginning of the 1900s, a drug only binds to a target if there is this kind of pharmacophore features there, right? This was Paul Ehrlich. It's not true, it was not, he said it in a different way. It came later on, it was Kier who said this, pharmacophores. But there's still something that is going on. A lot of things are going on. For instance, you can use pharmacophores to analyze MD trajectories. A big data amount that is very hard to analyze. It's very easy to transform this data into pharmacophore representation and then to look at the interactions. And this is an example here that our group did together with the group of Gerhard Ecker at the University of Vienna. One of his PhD students had worked on HSP90 molecules, and she had generated tons of trajectories, and we ran through these trajectories with our pharmacophore analysis tool and could draw an incredible amount of information out of there. And of course, first you need to develop a platform that is able to do this, right? To load in a trajectory, to transform this into pharmacophores, into dynamic pharmacophores, and then make the analysis. And so what we can see is here we can look at feature frequency plots, for instance. Here you see all the pharmacophore features that this ligand can make, and they are color-coded. Here blue is aromatic, yellow is hydrophobic, red is H-bond acceptor, green is H-bond donor. So you can see here the features, feature frequency. So for instance, H-bond donor is there all the time, almost all the time. Hydrophobic is almost there all the time. And then some of them are coming and going. And in this way, you can also get a sort of idea, what are the features that are really important for a compound to bind and stay very long in the binding site, for instance. Then we can look at unique pharmacophore features. So we can, because there will be pharmacophores that come and come and come again and again and again, right? And so we can count them, how many times such a pharmacophore occurs. And so we see here the unique pharmacophores with the number of features. We can, in a graphical representation, we can play with this threshold here and say, okay, now we have selected pharmacophores that at least appear around 130 times in the simulation, and these are these two models here, and these are the features that are visible, and so on. And so we can also look at which amino acids are doing the interactions with which pharmacophore feature. So this tool is even expandable to a sort of thing where we do hierarchical pharmacophore network analysis. So you can generate from such a trajectory a whole tree of pharmacophores and calculate the connections between them, right? Some of them do not appear at all, and so you need them to make some artificial models. But what we did here is, so this is the PhD work of Arthur Garon, who is now at AstraZeneca. He published a very nice paper of this hierarchical pharmacophore network analysis. And what you do is you look at all the pharmacophores that appear in this trajectory, and you make a connection, you connect them in a way that there are daughter and son pharmacophores from parent pharmacophores, or let's say kids and parents. And here you can, and we can see effectively, we looked at trajectories of activators and blockers of a certain kinase, and indeed, looking at this, we could differentiate pharmacophores for blockers and for activators. And so they differ in behavior and induce the formation of the inactive and inactive form of the protein because of the, and this becomes evident when you look at the pharmacophore network. When you use this pharmacophore network, then you can design activators or inhibitors, right? Very easy. And here you see again this kind of, there are two molecules here. This was starting from 1V4S, from the inactive form, this is the PDB code. The other one is 4NO7, the active form. Molecules look pretty different. And then they all share a sort of parent feature that everyone has. Then there are like the 2 feature models, 3 feature models, 4, 5, 6, 7, and 8 feature models. And you see here the activator is laying in the 8 features model. The inactivator here, the activator is here in this area. And when we look at these things, how they separate, you can see that one family goes in this direction and one family goes in this direction. And you can take then pharmacophores from here, for instance, or from here to get one property for the molecule, or take pharmacophores from here and here, or even here, when you do virtual screening to find the ones that would do the other thing. And you can understand why they do this, right? And you can design then even better molecules at the end. So this was this example here. So where do we develop this? So there are 2 ways how we develop. There's one prototype kind of toolkit that we developed that is open source and free to everybody. And then the other part is the software that we developed more than 20 years, since 20 years in the company Intelligent here that I founded with other colleagues 20 years ago. And this is called Ligand Scout, and this is now a fully integrated molecular design package with a high-end graphical user interface and command line tool, so you can do scripting also with this. And we have also KNIME nodes so that you can integrate Ligand Scout functionality into the KNIME workflow platform. It has a worldwide user basis, and it's known to be the leading solution for pharmacophore-based modeling nowadays. And we are still in next generation capabilities, active development, and 2 things that will come out this year is a way faster high throughput method for virtual screening on gigascale libraries, and also interactive pharmacophore-based clustering, and a lot of other things that are in the pipeline. But what is Ligand Scout? So this is this kind of thing and how has it been used? When you look at the literature, you find nowadays more than 3,600 papers where people did structure-based modeling, ligand-based virtual screening. They used it for hit identification, for fragment-based design, lead structure optimization, PPI elucidation, for drug repurposing, and for profiling. And you just look into Google Scholar and you will find very, very interesting papers where people use this software to find active molecules, and even molecules that go into clinics here. This is an example from Pfizer that they published a year ago. A compound where they had screened their library with a lot of methods and never found something. And with Ligand Scout, they identified a compound that they call compound 1 here in a specific kinase, and with 2 chemical modifications. So this is the original hit that was already around 20 nanomolar. So this was really highly active from their own collection. So they mastered the chemistry. Nobody else has these compounds. A little modification here gave already the lead compound 2.9 nanomolars, 3 nanomolars here. So a factor of almost 10 better. And then this went into development and got into phase 1, and I think it stopped after phase 1. And they just published a paper just a few weeks ago now with the results of the clinical study. So this is quite interesting, right? That you do have a very, very little modifications from the hit that you've identified, because your material that you have in your own library is already so good. This is the big advantage when you screen your own library. And another story where people in Japan used Ligand Scout Pharmacophores as a scoring function for a generative neural network to predict new compounds. So they designed and synthesized DDR1 inhibitors using a desired pharmacophore model, using a DGN model here. The way was that they used Campbell to train a network, and then this network was run through a pharmacophore that was built for finding DDR1 inhibitors. So starting from an X-ray structure, very easy thing. Then they generate molecules, put this into the pharmacophore, and each time the molecule fits to this pharmacophore, then the neural network gets a heads up, thumbs up, and learns how the molecules need to be designed to fit better to this model. And at the end, they had something like 20,000 molecules that were all pretty good. And then they did another filtering using Ligand Scout and the quick and dirty binding affinity score analysis, and synthesized the compounds. They synthesized around 10 compounds. And the best one that they found was a hit here that had, what was the IC50 was dual digit, 92 nanomolars IC50 for this new compound here. So starting from scratch, right? From a pharmacophore model with a DGN generating molecules and ranking them according to the pharmacophore. So these were just two examples, recent examples from literature. There's much more, of course, but I don't want also to talk about our own examples. What I want to highlight here is the thing that I really highly recommend you to download and to use for your own research. This is completely free. It's open source. You can also contribute if you like. This is the CDP kit from Thomas Seidel and Oliver Wieler in my group. There's a GitHub page. This is the QR code here. There's even, so you can even download executables to install. And you have also applications for a few functions, but mostly it is implemented in C++. It contains all basic functionality for cheminformatics. And it comprises the chemical data processing library, CDPL, together with command line software tools. So we have a CDPL C++ API and a Python interface layer. So you can write your Python code using direct access to functions that are here in this CDP kit. And we have integrated the major machine learning libraries like scikit-learn, PyTorch, and TensorFlow. So this represents our standard environment at the university group for prototyping novel algorithms. So this is where we test if things run well, we run it, and then if this is okay and we publish, then we make the follow-up implementation. We rewrite the code and put it into Ligand Scout and the KNIME platform, so that people who don't want to run a toolkit and do scripting can access it directly from a user interface. So the question is now, bigger haystacks, do you get better hits, right? So I told you it's to find the needle in the haystack. And I remember many, many years ago when I was young, there was a guy from industry who, in a conference one day, he stand up and asked, is it smart to make the haystack bigger if you want to find the needle? And I mean, this was the time of high throughput screening and people starting to do combinatorial libraries. And at the time, everybody was clear that making the haystack bigger is not a good idea. Well, this idea has been now taken up again, make the haystack bigger, because now we have this potential of having billions of compounds that can be synthesized, even they are not yet synthesized, but they could be synthesized. And so it's a business model of companies to announce they have like 40 billion compounds that they could make. So if you want to buy one, you can buy this, and they will promise to synthesize it. So if you want to do this with high precision, and you don't want to stay in the fragment space, which most of the people who address this problem do, and I don't believe that you will find the right compounds if you stay in the fragment space. I believe that you need to be more precise, and you don't need probably 100 billion of compounds. Probably 1 billion is enough to find enough hits to start a program. Remember, when you do a screening, a random screening, no, not a random screening, a high throughput screening with any library, you have a hit rate of approximately 0.1%. Sometimes it's a little more difficult, then you have 0.01%. Maybe it's extremely difficult, you have 0.01%. But if you have billions of compounds, the number of hits you get is huge, and nobody can test so many compounds then and follow up. So the question is, how many do you really need? So we believe that if you have 1 to 10 billion, you have way enough already. So the idea is to select the good ones, the right ones, to have this 1 to 10 billion. But still, we need to have a good, faster conformer generator and alignment procedure. And we worked on that. And I think now I'm very proud of this work here. Thomas Seidel was spearheading it, and it's published a year ago. This is our new conformer generator at the moment. It's, I would say, the best one that is out there for high throughput conformer generation. It's called ConForge. It's completely open source also. Can download it. It's part of the CDP kit here, and he can show that it's absolutely comparable with the best commercial products also, like OpenEye, and is for free actually. So it's based on our previous Icon conformer generator. It's available in CDP kit, and it's improved small compound performance accuracy, especially for complex structures such as macrocycles. It is integrated already in the alpha version of LiganScout XT, which is the next generation software, and it allows for faster database generation and possible better screening hits. It's more than 2 times faster than the old Icon fast. And in addition, in LiganScout, we have implemented a new LiganScout database format, which we call LDB2, with a reduced file size. 1 billion compounds currently are 16 terabyte, and so this is quite a lot, and we are still working on compressing this and putting this down a little bit. And together with a new alignment algorithm, which we call the G3PS alignment algorithm, this is the basis for Pharmacophore-based exascale virtual screening. It is running already. It's running in companies as prototype, and they search already the large data space. So this is this alignment algorithm that Christian Perman, one other postdoc in my group, published together with Thomas Seidel here, where we overcome problems that might occur sometimes when you look for RMSD or volume-based alignment. You would sometimes align like features that align perfectly very well, but then some features might not have a match. And so the idea, the desired alignment is this one, actually not this one here, when we have 2 pharmacophores that we need to align. And then it's also important to look at the exclusion volumes, where alignments sometimes would fail on exclusion volume sphere when they barely touch the exclusion volumes, which invalidates an alignment. But this, if you move it a little bit, it will not touch it anymore. And so this alignment algorithm is now bulletproof, tested, and what we do is an iterative improvement of heuristic guesses. So the people can use how many guesses they want. If you want a quick and dirty method, if you have so many compounds that you don't care to retrieve maybe only 80% of the hits rather than 100%, you would run very fast and you go maybe for 10 or 20 guesses. If you want to be precise, but a little bit slower because you have all the compounds in-house and you don't need to buy them and you want to retrieve all the ones that fit, then you go with a higher number of guesses. So it's runtime versus accuracy that can be tuned. And the found alignments are more accurate according to the user query and optimized for maximum number of matched features. The good thing is that you can also go for omitted features and it will not be much slower, even in combinatorial, right? So you can ask, you have a pharmacophore that has maybe 20 features, and you ask for a minimum of 7 features to be matched, and you will get maybe 10% slower than if you would ask for a full matching of a pharmacophore that has maybe 7 features or 8 features. And this is extremely important when you do these searches in apposite pharmacophore models that have a large number of features, because they have been defined based on the amino acid pocket, the pocket forming the amino acids forming the pocket that has no ligand inside. So, yeah, I don't want to go into too much details here with this alignment thing. I just want to mention a few other papers here where we used graph neural networks architecture to better predict standard molecular properties. We also invest in these methods now to test and see if they work better than traditional cheminformatics methods, and sometimes it does. And also here, we extend this to exascale library screening. Now there's one thing here, when we want to have a more quantitative description of molecules rather than qualitative, yes, no. how can we rank pharmacophore features in a pharmacophore model? Right? At the moment, when we do virtual screening, we only say yes, does fit, or no, does not fit. But how can we rank molecules? How can we have quantitative models? And I don't want to speak about structure-based modeling. We have some ideas to do this using geometry of features, for instance, or MD trajectory analysis. I just want to touch a little bit on this ligand-based modeling, where we explain quantitative differences in binding, like expressed in Ki or IC50, for instance. And we have started based on a method that we called GRAIL to generate, and I will explain this GRAIL in a minute, to generate ligand-scout merged pharmacophores to select features, to do cross-validation in combination with biological activity data, and then find the features that are relevant and weigh them with a certain correction factor to get, at the end, when we match compounds to these features, a quantitative value. And we named this QFAR, for quantitative pharmacophores. And this is the paper. This was a PhD of Stefan Kohlbacher. And it's not only the algorithm that is described on GitHub that you can download and play around, but it's also implemented already in KNIME workflows that you can directly run. So how does this work? You have training steps, like 4 training steps here. First, you have the training library to select a template out of this, which must be the most rigid, most active molecule, obviously. Then you do in the step 2, you align all the possible pharmacophores of all other ligands. You have a pharmacophore feature container, then you do a clustering, take the centers of the clusters, put the features there, and then you have a post-processing step where you learn with a method that is able to deal with a low number, 20, 30, 40 molecules, maybe 50 molecules, like random forest, something like that, or support vector machine, to generate then this quantitative model. And what we do is a decision tree here, a decision tree method for the training. So we calculate the distances, basically, of the optimal feature to the feature that we observe for all the ligands, and then we can train this with that as a decision tree and make a regression model where we can, here for instance, plot the actual versus the predicted values. And in the cases we tested it works really well so far, so we wait for more applications where people play around with this. So this is the application of these quantitative pharmacophore models within the work of a neuro-de-risk project, where we made quantitative models for toxicity, actually. And we have also an automated workflow that is in KNIME, where the things are automated, where you only have to present the data sets and make some parameters at the beginning, and then the whole thing runs automatically. And this is the paper for that one, the KNIME nodes implementing the QFAR algorithm. And this is what it looks like. So you have like 4 nodes. You have a QFAR learner, a QFAR predictor. You generate your models, and then when you have them, you can read them in again. And of course, when you have generated a model, you need also a QFAR writer that will save your model. And this is such a workflow that is provided for training and running these calculations in KNIME, right? So you only need your molecule data sets with the activity values. And this is, for instance, a usage here where we used quantitative prediction of drug-induced seizure risks using a quantitative GABA-A model. So GABA receptor, the GABA ion channel, is when you block this channel or you negatively modulate it, it can induce seizures in your brain. And this is one of the big problems in early drug discovery. You don't want to give compounds that might induce seizures to patients in phase 1, for instance, right? This is something that is very difficult to predict. We have now some quantitative models. We have qualitative models that are implemented also in industry to look at the risk of molecules to induce seizures. Once such a molecule is flagged, of course, you can go into animal experiments or like zebrafish models, for instance, to check if this is really the case or not. and at least you get a sort of idea of a risk. Another thing that we are just published, and it got accepted with some revisions, and we just submitted the revisions, so I'm happy. I hope that next week or so we will get the final acceptance, is the work from Christian Fellinger, based on GRAIL again, and I will tell you GRAIL again in a minute, deriving descriptors from these GRAILs, and being able then to use these descriptors. These are pharmacophore-based descriptors, actually, of compound in a complex with a protein, taking into consideration the interaction probability from the protein, taking into consideration the interaction probability from the ligand, and transforming this into pharmacophore interaction regions, these GRAILs here, and then taking these descriptors together with some other descriptors, which are scalar descriptors like log P, number of rotatable bonds, etc., and train this in a deep learning method with the PDB bind, for instance. And Christian could show that with these descriptors, he's able to pretty well reproduce the binding affinities. And now the idea is to take this, to use this as a scoring method for docking results, for instance, or for pharmacophore-based virtual screening results. And it looks quite good, and it's extremely fast, faster than other scoring methods. Now, what is GRAIL? GRAIL is this thing where we it's the abbreviation for grids of pharmacophore interaction fields. So this paper is a little bit older already. It was first published in 2018, so the concept and the first application. And what we do here, we use this to look into binding pockets and characterize binding pockets according to their, I would say, pharmacophore trend, right? What kind of pharmacophore interaction would this pocket look for? How must the ligand look like that it fits well into this pocket? So what you need to do is you look at the amino acids around, and you look at pharmacophore features that could be done from the amino acids. So it's a sort of APO pharmacophore model that you generate, and we do this technically, that we put a grid into the binding site, and on each grid point, we apply the ligand scout pharmacophore heuristics. So for each feature, we have kind of rules where we know if this point is close to amino acid that has these and these features, then the probability of having a pharmacophore there has a certain value. So for each grid point, we have a certain value. And then we can look at all these values and contour regions that have the same kind of wish, for instance, for a hydrophobic feature, or for an H-bond acceptor, or for an H-bond donor. And the good thing is we can do this not only on one X-ray structure, but we can also run this on MD trajectories. So we have a complete description of the binding site in pharmacophore description for MD trajectories, not looking at the ligand. Of course, if the ligand is there, we can see what the ligand takes from the interactions that are possible. But there are more interactions there, right, that could be done. So the ideal ligand would make more interactions, would make all interactions that the amino acids want to have. And this is the idea behind. So we have distances, of course. We have angles that we need to take into consideration for directed interactions, like H-bonds, for instance, or pi-pi stacking. So it is fast, so it's amenable for the analysis of entire MD trajectory. So it's not on the second, right? But it takes a few minutes to calculate such a trajectory description, interaction description. And the script is also available in CDP-Kit, so you can run and play this and run this yourself also in your own lab here. And I just want to show you here in this HSP90 case study that Doris Schütz made what it looks like. And so we look here only at certain amino acids that are known to be important, and we look at the feature regions that cry for certain interactions. So here, for instance, in A, you have aromatic interaction. So the binding pocket is on this side here. This is the rest of the protein. So we see here, this would like to have aromatic. In B, we see the hydrophobic part. In C, we see the positive ionizable proton against the aromatic. So also an aromatic ring might want to have a proton sitting, a positive charge sitting, not a proton, a positive charge sitting next to it because of the pi cation interaction. Then for D, for instance, we have here charged amino acids, lysine here. And of course, lysine wants in a certain distance and distribution a negative charge around it, and so on. And so you can look at all these different interactions, and at the end you get an entire picture of the potential interactome of a binding site. And you can use this in lead optimization, of course. You can use this because you easily understand the medicinal design, chemistry design guidance that needs to be done. You can focus on specific regions. For instance, you can look at water molecules. Each water molecule in the trajectory, you can look, is this water molecule sitting in a favorable region? It means, is it entropically favored? Or is it sitting somewhere where it wants to get out desperately and you need to replace it by a ligand part, right? Entropically defavored water molecules or unhappy water molecules. And if you know this, you know where you need to position your little methyl group or a chlorine, for instance. So this magic methyl story, you know, when you do at the end of the optimization, you try different positions of your molecule, you put a methyl group, and then all of a sudden you get an increase in affinity by a factor 10 or even 100 because it fills a little hole that displaces the water molecule, pushes it out, and makes the binding stronger, which medicine chemists know as magic methyl. And also, of course, you can do this pharmacophore hotspot feature frequency analysis for prioritizing replacement modifications of molecular substructures and give this interaction preference guidance. And of course, what you can do is it's easily adaptable for generative de novo design. So plug in a generator of molecules into this, and it will make molecules that fit exactly, after a while, after it has learned, it will make molecules that fit exactly this kind of description of your binding site. So back to the haystack now. Again, is there a way to use high-quality pharmacophore information but avoid this conformer generation and 3D alignment? And we just have submitted a paper that has been accepted in a conference by Daniel Rose will present this paper. And what he did is he generated pharmacophore vectors that can be used without alignment anymore. So you can do virtual screening without alignment using pharmacophore vectors. So the idea, the original way, the traditional way to do a virtual screening to get a hit list is to generate a pharmacophore model, for instance, starting from a PDB structure or from an MD trajectory. You get a pharmacophore. Then you have your ligand database. You make the conformation analysis. You calculate all the pharmacophores for all the conformations. And then you do the alignment and you get your hit list. But could you do this differently? So he had the idea to do the same thing, to create a ligand database, make a conformation analysis, and encode this into vector representation. Then you have the pharmacophore model, of course, that comes again from an X-ray structure or from a ligand-based model, encode the same in a pharmacophore vector representation, and then use this embedding space property filter to find a hit list without alignment anymore. And the idea behind is this kind of REX, what is called the retrieval of concepts in databases. This is a method that uses vectors to retrieve a concept in a database. So for instance, when you have these vectors, and it can be used in search engines, in recommender systems, and of course in chatbots. So this is what we all now face when we use ChatGPT or Gemini or so on. So let's explain it in a very simple way here. We have vectors that point to certain it's concepts here, computer science, data science. Of course, they are very close to each other, right? Because computer science and data science are close. When you look at molecules, this is somewhere else. Caffeine is a molecule, so it's close to molecule in this property space. And when we look for cheminformatics, it should be somewhere between data science and computer science and molecules, right? So this is the idea behind. to have vectors that point in directions. And the idea is now how to train such a model, such an embedding model. And Chris and Daniel came up with this contrastic loss function that can be used in a Siamese network where we compare things and we then make shared weights and adjust these weights. And so a positive pair, for instance, would be computer science and data science. So we maximize the weights here. If we are divergent, we would minimize the weights. This is called contrastive learning. So the question is, how can we use this? What shall we compare to train a pharmacophore embedding model? And the idea here is then to use, again, subgraphs and supergraphs and use the subgraph isomorphism for order embedding. So we have a target pharmacophore here. We have a query pharmacophore. And we can look at what is the supergraph of a subgraph here. So this subgraph would be part of this supergraph, but there can be other things in there, but we need to have these things. And when we look at these embedding dimensions, we can then define a so-called target region where the pharmacophore vector that is used for this embedding is in the target region. And all the pharmacophores that are in the vector are represented in these target regions are interesting ones. Some that are off target will show up somewhere else, right? When we project this here, it's only 2 dimensions, but for understanding the concept. So we look always into this region here. And so what we do is we have this query target pair, the target graph and the query graph. We do the encoder again, embedding. We do the shared weights. We get the target embedding, the query embedding. We do the contrastive loss. And at the end, what we see here, for instance, in the pharmacophore, we have the subgraph, we have the supergraph here, and we have another graph that is laying somewhere else. So we know exactly which graphs need to be in this region. And it was tested on a first data set here, an arbitrary pharmacophore model, and the drug-like ligand database. 1 query, almost 4,000 ligands, less than 25 conformers. And so this was the representation. So a lot of vectors, of course, here that are not in the region, but these are the ones that are the interesting one because the target pharmacophore that we are looking for is located here. And when we see this, it takes a little more space on the hard drive, of course, because we need to have these vectors encoded again. But the screening time is 100 times faster. And this is not yet optimized. So when you screen in CDP-Kit, which is extremely fast already, you have this in 2 seconds. And with this vector match, we have this in 200ths of a second, the same result, right? So now we believe that we can use this also for ultra-large scale virtual screening. And there is still optimization possible. Potential for faster database search is to use not a normal database, but a vector database for faster retrieval, and to use the inherent tree structure that is in this pharmacophore representation there to go faster than using a traditional relational database. So another application for pharmacophore vectors could be also the nasty problem of hydrophobic perception. This is something pharmacophores work very well in the area of directed enthalpic interactions for H bonds, for instance, but it's a little more tricky in the entropic case, and here hydrophobicity plays a role. And we think that we could do the same thing with vector embedding of a point cloud, but having not the classical hydrophobic features, but atom-type hydrophobic features, and then use this point cloud that is much bigger here in a vector, and use this for the definition of hydrophobicity for virtual screening in this pharmacophore vector format. But this is still future. This is our idea, and we want to work on that. And I want to close this lecture with a few examples here where we really used our methods to help scientists to develop better compounds void of risks of toxicity. Because, you know, I mean, one thing is to have a good molecule that binds to the target, but it should be selective, not bind to off-targets. And off-targets that are related to toxicity that are difficult to predict in another way. And neurotoxicity is such a thing. There is no way to predict, no good way to predict neurotoxicity. And so we got granted a European IMI project a few years ago that was initiated by pharmaceutical industry. And we called it neuro de-risk, to de-risk compounds for neurotoxicity. And the idea was we had to de-risk for seizures, we had to de-risk for peripheral neuropathies, and we had to de-risk for depression and suicidal ideation. You won't believe how many compounds are there that induce depression. Drugs that induce depression, drugs that people take, and even drive them into suicide. This is a problem. And of course, you don't want this when you put a new drug on the market, right? It's hard enough already if a drug works well, but still people commit suicide. But a lot of other people have a benefit of it. It's maybe not so easy to take a decision to take this off the market. But if you have a new compound, you can definitively do better. And so there are two approaches, of course. For seizure, for instance, we have a clear target-based approach because we understand very well how seizures are induced in the brain, right? And GABA is one of this target here. And so we have different things here that we can look at. We can do structure-based modeling. We can generate pharmacophore models. We can generate quantitative models. And if a compound hits this model, then the chance that it's inducing a seizure is high. And we just take it out. Or if the compound is very, very important, we test it at least in animal models. For suicidal ideation, for depression, it's more difficult because there is no clear mechanisms yet. But there is now already one mechanism that is elucidated. We know, and there's a company in France, in Montpellier, Yesylevskyy, who did find out that some drugs that clearly drive patients into suicide are modifying receptors for serotonin by post-translational modification of RNA for these receptors. So they drive up the modification of the RNA that then translates into mutation of the receptor biochemically. Right? So there is a biochemical reason why people kill themselves. And we know the compounds that do this, and we can make so-called outcome-based models. Again, translating the structure into a pharmacophore and doing this. And so what we did is we created for seizures, it was easy. This was structure-based modeling mostly, a little bit of ligand-based modeling. For this suicidal ideation, it was more tricky. We looked at pharmacovigilance data. We collected all the compounds that have been reported with this. We clustered them according to pharmacophore space. We generated models. And then together with Yesylevskyy, we checked these models, whether they are correctly predicting this one mode of action, this RNA editing. And yes, we kept those molecules, these models from molecules that were correctly predicting this. And we put this into the toolbox. So now, this neurotoxicity toolbox that we call neuro de-risk toolbox contains 60 3D pharmacophore models that have been so far incorporated and developed. And so you see the receptors here, AMPA, NMDA, suicidality, GABA, glycine receptor. Then we have also models for BBB transport, of course, and peripheral neuropathies. And they are all built in a so-called neuro de-risk profiler node in NIME, where you can just funnel in your compounds. You can draw them with an editor here in NIME, or you can load an SD file or a ligand scout database, whatever. In this node here, all the calculation is done. All the models are in there, and all the molecules that go through are tested on all the models, and at the end you get a sort of heat map here. You can visualize a heat map and see what molecules hit which target pharmacophore. And the ones that are completely clean, they are white, there is no thing, and the more red it is, the more dangerous it seems to be, right? So here you can see the molecules are on the top. So for instance, this molecule here on the far right only hits one model, but it's not, it's only orange, so it's not red, it's less pronounced. Whereas, for instance, this molecule here, the first one, this hits 5 targets with a very strong signal. So this would not be a molecule that I would continue to look at, right? But so you can get an idea. And what you can do is you can play around with your molecule in this 2D editor, run it again, and see if the profile changes. And then this helps the medicinal chemist to understand what are the features that he needs to avoid to get this outcome. So, I want to come to the conclusions of my lecture here. I strongly believe that these next generation pharmacophore models are ready for the most difficult challenges in computer-aided molecular design. They are highly useful for hit ID and prioritization, and of course lead structure optimization. They are amendable for high-precision virtual screening in exascale libraries, and exascale now is like, for us, is 1 to 10 billion of compounds. They are most comprehensive tools for medicinal chemistry guidance. So a medicinal chemist that uses these pharmacophores understands why she or he needs to change the molecule. And we can take into account dynamic effects in binding kinetics and translate this directly to design. And with this neurotoxicity stuff, we can get better lead structures void of toxicity risks. And with that, I want to thank you for your attention and to invite you to 3 websites here. The one of my group here in Vienna, ChemInfo, Cheminformatic Group at University of Vienna, then the CDP kit of Thomas Seidel and Oliver Wieder of my group, and the company website, intelligent.com, where you can register for evaluation license and you get a free evaluation license, and you got already maybe from Karel also the download links. Anyway, they are installed upstairs, and you have also already a license that runs until, I think, end of February to play with this. And with that, I'm ready to take your questions. Thank you. Yes?
Can we go to the slide where you're talking about the GABA-A receptors and drugs which are developed against seizure?
This.
Yeah. I think even before you were talking, there was GABA-A.
Wait a second. This one. Yeah, maybe I wasn't. So what? It's just to be clear. We are not looking for drugs against seizure. We are looking for drugs that do not induce seizure, right? So, and maybe this is what you wanted to see, where all this here, right? Maybe this is what. So all this, we know that these are, on one side, they are.
There was even before.
Even before?
There was with some names from zinc, I think.
I know. The quantitative models you mean. I think I know what you mean. The ones that we did for, these are the assay numbers from Campbell. This one, I guess. Yes. This is the assay ideas and the number of molecules that we used for the quantitative models. So there are some quantitative models in assays in Campbell that we used for seeing if how well the method works, and for quantitative modeling.
And these are all models with the GABA-A receptors.
I would say so. I'm not, honestly, I don't know for sure now exactly what, but I guess that they are all somehow related with GABA-A, yes? But they could be agonists, could be blockers, could be positive allosteric modulators. You know, all these.
That's what my point is. Usually you have no clue how does it react, because there are so many points of interaction between the
Absolutely.
chemical and the GABA A receptor.
Yes. Yeah, exactly. So this is, you're absolutely right. This is the most difficult thing because there's so many binding sites. But now we understand more and more because we have two different things. First of all, we have people in Vienna who are very well, I would say, they know extremely well the whole pharmacology, and they know all these binding sites. Now we have also the cryo-EM structures already. And the other thing is we have a lot of data from industry. They test their compounds in certain assays. And of course, it's much more complex because we know that the constitution of these gamma receptors is a heteropentomers, right, with alpha, beta, gamma subunits at different 1, 2, alpha 1, 2, 3, beta, etc., etc. So it's very, very complicated. But we rely here on assays where compounds have been measured in one specific assay. For instance, this Campbell 1273617. This is an assay where they have tested compounds on the same assay and get some data. And with this data, we model an effect. And we try to quantitatively model this.
44 molecules and on that.
Yes. But again, this is very good question, yes, also because normally when you do deep learning also you need big data, right? But this is not deep learning. This is, again, this is a decision tree method that is used for selecting the features in a way that they make the most sense. So we can't deal with this low number of compounds.
But even with this one assay, is it known that this assay is done on this binding site or is just by effect?
Honestly, I cannot answer this. I cannot answer this. But this is a very good point, of course, yes. Still, I think there's 44, in this case, 44 molecules. And the only thing I can say is that we have, for these 44 molecules, we have a pharmacophore model that reproduces the activity of these molecules or the value that is measured of these molecules when you run them through the model. And we expect that if you run molecules that are not related at all, if they fit to this model somehow, then we get also a quantitative value. And again, this is what I said at the beginning. Sometimes you don't care if the model is based on the right binding site. As long as the model predicts the right thing also for a new molecule, it's a valid model that you can use then for de-risking other molecules. Yes, Martin?
So Thierry, thank you for this great talk. I have been following your talks on Pharmacophores throughout the years, and I'm always fascinated how you bring it to new levels and directions, etc.
Thank you.
I have two directions as a further future, maybe. What is your opinion about that? First is that, if I get it right, you have the different features, and you take them as the same, let's say, contribution to the affinity. So one thing is whether you would try to give weights, whether some are stronger contributors. And,
yes, exactly, yes.
And then second, you rely in the dynamic pharmacophores on ND and hoping that the results are correct. And what is becoming clear lately that charge interactions, salt bridges, are overly strong, overbinding. So again, there would be like, we are working on some scaling down so that they don't appear so often. So that would be like for you as an input, and that's one option. The other option is going to these weights, and maybe like if you see them too much, then like weight them down.
Yes, absolutely, Martin. Very, very good points. So in the ligand-based modeling again, so this is what we try to do, to put weights on these features, which is, I mean, there we clearly need some algorithm to analyze the data and correlate the data with these features. But as you said, and this is here what I did not touch on here in this lecture, but we have also ideas how to use these feature frequencies, for instance, in an MD simulation, to try to correlate it to affinity maybe, but affinity is one thing, but to residence time. You know, there is always this discussion, what is more important? Is affinity more important or residence time? Somehow it's correlated because something that binds strongly will stay longer, yeah? But sometimes a slow K-off is, for instance, the thing that you want to have. We believe that there is something behind there that we need to find. And I absolutely agree with you. What the problem is at the moment is that these MD simulations are run on force fields. And the force fields are not ideal. We know this, but this is the best we have at the moment. And you're absolutely right. I mean, electrostatic forces are overestimated. Yes, absolutely. I will be happy to see maybe large language models applied to MD simulations where this kind of things can be scaled down. Maybe it's something for the future. Or even things with quantum mechanics or you know, this DFT methods for MD simulations would be great to have this. I mean, maybe in 10 years we have it. You know? Looking at the development in computer science and in AI, I mean, I think the problem is also still having good quality training data for this, and you know it yourself as the expert in this field. But I think even with people like you contributing and other people in the DFT field and the SQM field, I think this is, yeah, we will get better. But I agree. This is the way to go. Yes. Other questions? Sergey looks, you do have a question?
Yeah, of course, I have a question. Yeah. Thanks, Chair, for this wonderful talk. I'm really excited to hear how we can use this ligand scout for, especially for our malaria project, for example. And my question is about reinvent, because we also plan to use reinvent. And reinvent provides you smile strings, so you need to convert them to 3D conformations. Do you provide an API for ligand scout?
Yes. Yeah. This is both in ligand scout and in CDP kit. You just read in the smiles and it makes automatically a 3D structure out of it.
And next question also about conformer generation, because maybe something missed for me. Because so what I typically know about conformer generation is that you use some kind of force field, for example, universal force field, and you just do force field of quantum chemistry optimization in vacuum. But also you can do it in water, or some kind of induced effects for ligands.
This is a very good point, actually, and it comes also to what Martin said here. So this, you are right, of course, and it's pretty artificial, but we still have these force fields. But you can scale down again the electrostatics, because when you are in water, for instance, you have like a factor, the electric constant is like 80, right, of water. So what shall we consider as a dielectric constant? Shall we consider 20 or 80? You know, so what we need to do is look at the microenvironment of the protein. And we have programmed our force field in a way that you generate a number of conformations that is, I would say, manageable. So we have like settings for fast setting that typically creates like 25 conformers for a small organic molecule. And we claim that in these 25 conformations, also the bioactive conformation is there. And we have tested this. I mean, Johannes Kirchmeier has written a few papers on that. He has a data set where he has this collection of bioactive conformers or he collected this and used this for testing force fields. There are two papers, conformer generators. There are two papers from him where he did this. And yes, both with Conforge, but also with ICON, we are confident that within the family of conformers that we generate, the bioactive conformation is there. Maybe not the absolute minimum conformation, but at least the bioactive conformation, which is what we want to find, right? And it's pure brute force, fast, ultra fast molecular mechanics. Unfortunately, this is what we do.
With kind of customized parameters.
Right. But feel free. I mean, you have it. And honestly, I never got any complaint from the conformers generated. And the idea why, maybe let's make it very clear. It's maybe not completely precise, the conformation that you want to see. Maybe there's some little 0 point some angstrom that is not ideal. But we have this fuzziness in the pharmacophores. And so what we do is we try to retrieve a compound according to the features. And depending on the size of the features, you can go when you make them small, you can go very like straight and very selective and retrieve less. And when you make them a little bit bigger, then of course, if you have a wrong conformation, it will not be good. But if you have the standard size features, which are 1 angstrom diameter, we know that we find the bioactive conformation for sure. And we can test this by just taking all the pharmacophores from the entire PDB, take all the ligands from the PDB, flatten them down, export them as smiles, regenerate them, and run them through, and you will see it works. Yes. Great. Are you looking forward to the hands-on? Oh, yes, you have a question.
Thank you for the talk. The question about the QFAR approach. And as I understand, you used a quite small number of compounds, and it was single-task prediction. And the question is it possible to make multi-task prediction model for specific family of targets? Because normally for this, compounds will be similar, and the matrix will be quite compact and informative. And maybe the share of information between targets.
Yes, it's a very good idea. We didn't do it. Yet. There's only a few things that we can do. There's a lot of. Come to Vienna and we do it together. Yes. Good idea. Okay. So it's very exciting, because there's so many things one can do, and we still are limited in our resources, in our time, but it's very exciting. So we are not getting bored. Okay, so if there are no more questions, then we will have a short break, and then we will meet again in the lecture hall upstairs, and we will play, and you will have fun, hopefully. Okay, thank you.